Good morning, ma'am. Good morning to everyone present here. I am Ujjal Sanyal, Assistant Professor in the Department of Commerce, Maulana Azad College. And this is my pleasure to welcome you to this morning session of online refresher course on Bioscience, Bioengineering and Nanotechnology, organized by UGC MMTC University of Calcutta and hosted by Maulana Azad College. We are honored to begin today's academic session with the lecture by the distinguished scientist whose work has greatly enriched in the field of virology. Dr. Mamta Chawla Sarkar, Scientist G, Division of Virology, ICMR, National Institute for Research in Bacterial Infections, Kolkata. Dr. Mamta Chawla Sarkar is a Senior Scientist G at National Institute of Research in Bacterial Infections, ICMR, Kolkata. She is an accomplished researcher who has spent her career studying how certain viruses, especially the rotavirus, which affects the children, interact with the human body. Her work helped scientists understand how viral infections develop and how they might be controlled. Her achievements include fellowship in the National Academy of Science, India 2013, National Women Bioscientist Award 2013, Kanishka Oration Award 2017, fellowship of the West Bengal Academy of Science 2018, fellowship of the Indian National Science Academy 2020. With over 100 research publications and impactful work spanning viral genomics, host immune pathways, and therapeutic strategies, Dr. Sarkar remains an inspiring figure in contemporary virology research. We extend a warm welcome to all participants joining us in this morning session and look forward to an intellectually stimulating session. Please feel free to share your questions in the chat box. We will address them during the session segment the following the research. Ma'am, the floor is yours. Thank you, ma'am.
Thank you. Can you hear me?
Yes, ma'am.
Okay, thank you for inviting me to this. And I hope that I was not sure about the audience. So I have tried to keep it very basic, the information, so that people at least understand what is virus and what needs to be, how to detect and stuff. So this, it's not too much of complicated science. So I hope that I will also encourage the participants to ask the questions if they have any query. If I know the answer, I will tell it.
Definitely,
So,
ma'am. It will be very engaging session.
okay, so I think should I start?
Yes, ma'am. Yes, ma'am. Thank you.
Okay, okay. So can you see the screen?
Yes, ma'am. It's visible.
Okay. So good morning to all the participants. And I think I'll do the introduction session in the end because I need to also know their background. So I will just first initially start with the presentation. So today I would be giving you a very basic introduction to virology, from diagnostics to what and how we can do research to control viral infections. So first is, what is a virus? So I hope most of you, because if you are from the biological background, you would know that it is an obligate intracellular parasite, which is made up of nucleic acids, DNA or RNA, and wrapped in a protein coat called a capsid. It has no nucleus, no organelles, no cytoplasm or cell membrane. So it is basically non-cellular, but it is living because it once it infects the host cell, it can replicate, it can divide, it can make multiple progenies. So, but it does not belong to any kingdom because of its intermediate stature of living and non-living. So why should we study viruses? This is the first thing. The reason is because even though we think that viruses only cause disease and when we are sick, and the doctor tells us that we have virus infection, we think that we have been infected with viruses. But the reality is that we live and prosper in a cloud of viruses. They are there in all, in like every time we are breathing the air, we are inhaling thousands of microorganisms, including viruses. They infect all living things, prokaryotes, vertebrates, plants, etc. And we also carry viral genomes as part of our own genetic material. When the full human genome sequencing project was done by NIH in US, it was found that the actual human genome, what we say is a human genome, it constitutes only 36%. Rest of the genome we have, which has been integrated into us from various pathogens and like viruses or bacteria or any other pathogens which have integrated into our system over the time and evolution. So they can cause variety of diseases from common cold and flu, which we in at least Calcutta we suffer all through the year, to serious illnesses such as AIDS, dengue, fever, measles, smallpox, bird flu, and then COVID-19. So we also need to learn about its biology, structure, and also how to know and develop vaccines and antivirals for the prevention and treatment. So I would just like to also tell the audience that there is a myth or there is a concept that all the viruses are very very harmful, but it's not always like that, because since I told that we are getting infected all the time, but we do not fall sick all the time. So it is a no virus because it's an obligate parasite. So parasite usually does not want to kill its host. That is the first uh, part. Because if it kills the host, it cannot spread. It will also die. So therefore, it is the disease which we have or the sickness what we have is actually an outcome of how viruses has infected us and how our body or our immune system is reacting against it. So this it's a combination of both which results in the uh, what do we say outcome or the disease. There are people who are carriers who get infected but they do not develop symptoms. So the host has an important role also in the outcome of the disease. So how old are viruses? There's estimated molecular evolution said that viruses were even present in dinosaurs. If they have been traces of their genome found in the fossils, and so it is like they likely originated billions of years ago. That is even before the unicellular organisms developed. These are the ancient references to the viral diseases. We can see since we do not have the text, and mainly during at that time people did not know what was a virus or what was a bacteria. Anything or the diseases which happened were considered as curse of God. So, but based on the wooden carvings and the excavated material, there has been diagrammatic representation which have shown that like the wood carving showing a rabid dog people are trying to kill during the Egyptian time. Then also in Mexico, if there was older paintings which were found which showed that smallpox even existed in 16th century, because the representation of the picture clearly shows it as a smallpox. Then these are the Egyptian in the Egypt, these structure like the stone carvings have been found which show that there was polio was present in the 18th century also. Similarly, evidence of smallpox in Egyptian mummies have been found. So we know that there were diseases in during ancient times also, though people did not know it was due to virus. So the human beings we see have been largely threatened by viral diseases in the last 2 centuries. Prior to 1890, most of the deaths or most of the outbreaks or the pandemics were due to bacterial infections. And the major one of the reason is because that time viruses were not discovered. So see, we had plague, we had Black Death of bubonic plague. These were all before the 1300 or 1500 uh, century. Then after 1890, we have been seeing the viral pandemics like the Spanish flu in 1918 and 19, which caused 50 to 100 million of deaths. This was caused by influenza A virus. The Russian flu in 1889 to 90, 1 million deaths. Similarly, Asian flu, then Hong Kong flu, and then we had HIV pandemic from 1981 onwards. And then we have COVID-19 in 2020, Ebola during 2014 to 16, similarly, MERS, swine flu, etc., etc. So the reason why we actually started seeing more of viral outbreaks and pandemics after 1890 was because they were actually, people started differentiating between bacteria and virus. And first it was in 1892, Ivanovsky found that there was something which was not uh, culturable. It was not cultured in the media, but it caused infection. And similarly, in 1898, Bejernik called it Contagium vivum fluidum. The name was given because the the infected material did not grow like bacteria in the media, but whenever they infected the animal with that, the disease was there. Then in 1898 only, Loeffler and Frost found out agent of foot and mouth disease, and they found that if we filter the thing, but and then we get the supernatant, we still can infect the animal with that, suggesting that it is filterable. Like the bacteria gets blocked in the filter, but the viruses will pass out because they are very small inside. So the key concept that they developed was that agents were not only small, but they replicated only in the host, not in the broth. But the name was not given at that time. So since then, we have also, in spite of having a lot of pandemics, a major upsurge in the viral research and viral diagnostics happened in the last 50 years or so. And we have seen, therefore, we have seen massive upsurge of diagnostics and research in viral we are having antimicrobial resistance, but for the last few decades, the bacterial infections were under control because of the newer and newer antibiotics which were discovered. But for antivirals, we still have very, very limited antivirals which are available. Similarly, the vaccines are also available against very limited viruses. So early diagnosis is the key to avoid the mortality and the spread of infection. Because if we can diagnose early, we can actually contain the patient so that the patient does not spread it to other people around him, and also proper treatment can be given to avoid the death. So the lower and upper respiratory tract infections are caused by viruses such as influenza, RSV, corona. They are the second most common cause of adult and pediatric morbidity and mortality worldwide. Viral diarrhea is another major cause of childhood death, and rotavirus alone has been estimated to cause around more than 100,000 deaths per year. So if I look at this slide actually is only to show you what is the difference between the terms endemic, epidemic, and pandemic. When we say something is endemic, like in our country, we have rotavirus as endemic, dengue as endemic, because it is there, it happens every year. People get infected during the season and then they recover, and then there is a period in which that virus is not active, and then again next year again the disease happens in a small pocketed areas, and then again people recover. So that is called endemic. Like it is surviving in your country, it is part of you. It only comes up, the peak goes up and peak goes down based on its season. Epidemic is something when suddenly there is a change in the virus. It can be due to mutation, it can be due to a jump from animal to a human, and then the population does not have the immunity, pre-developed immunity against that virus, and then suddenly there's a lot of cases and hospitalization, and it spreads within the state or within a country. Then it is called an epidemic. However, and the next stage is pandemic. Pandemic is something when within a short period, maybe within a period of 1 month or 2 months, the same virus is spread across the continents. Then the term pandemic is used because it has it has become pan epidemic. Pan epidemic is multiple countries are having epidemics, so it is called pandemic, which we have seen in 2020 for COVID-19, as well as if you people remember, I don't know what is your age, but in 2009 we had swine flu pandemic, which was influenza A H1N1. So why COVID-19 was called pandemic is because it was first case was recorded in 2019 of 8th of December in Wuhan, and by the time it was uh, October of 2020, it was all across the world. Actually, it was all across the world in February only, but by in March, the WHO defined it as pandemic because it had spread to more than 5 continents. So this is the basic difference when we say endemic, epidemic, and pandemic. Our country, because it's a tropical country and we have a very large population, so we actually have many. We are endemic to many viruses and bacteria. Say viruses like dengue, rotavirus, influenza, also chikungunya. Then, if we look at the bacteria, then cholera, typhoid, parasites, malaria, we are endemic to it. So, what is the nature of viruses? Viruses nature first thing is that it needs a living cell to survive. We will only call it a virus if it requires a living cell to survive. It is released inside the cytoplasm of the host cell, and the genomes are made of either RNA or DNA, not both. It will not have both RNA and DNA. It can be a single stranded RNA or a double stranded RNA or a double stranded DNA or genome. And it is always packaged in a protein coat. The genome is packaged in a protein coat. The basic rules of virus architecture, structure, and assembly are similar for all the families. They come and form many geometrical shapes, and they come in various sizes and shapes. But the basic structural features are determined by the requirement for the assembly, exit, transmission, attachment, and other functions of the viruses. The capsid protein is the basic unit of the structure, and functions that may be fulfilled by the capsid proteins are to protect the virus nucleic acid. And the capsid protein is the one which actually activates the immune system. So it is also the immunogenic part of the virus, because that is what the human or the host immune system sees first. It protects the virus nucleic acid. It interacts specifically with the viral nucleic acids for packaging, interacts with the vector for specific transmission, interacts with the host receptors for entry to cell, and it also assists in the process of virus assembly and exit from the cell. See, the viruses are very basic structure. They do not have too many of the enzymes like what we have. They use the host machinery, replication machinery for their own replication and transcription and translation. Most of the viruses have a RNA dependent RNA polymerase only because they have RNA genome, and most of the hosts have DNA genome. That is the only enzyme they code for. Rest, everything they use from the host. So they have the genes which encode their proteins, like for their virus structure and the multiplication of their genome. That's all. They do not have any other complicated genomic structure, but they do need to have a capsid protein. So their gene, they have genes which code for this structural proteins to form their structure. So if I look at this example, this is the sizes from 17 nm to 3000 nm in diameter. This can be a rod-like structure or a circular or a geometrical structure. This is the outer layer is a capsid protein, and it is symmetrically organized. And this actually forms a 50% of the virion. And the viruses can be either enveloped or non-enveloped. That means they can be an envelope on the inside of the capsid protein. So if you look at this, this is a naked virus. This only has a capsid and a nucleic acid. But if we look at this, it has the capsid as well. It has an envelope around it. So there are two different types called naked virus as well as an enveloped virus. And I already told about the genomic material, that it's RNA or DNA. So this slide shows you a structure compared. This is a human DNA virus, RNA viruses, and in between is a bacterial viruses called bacteriophages, MS2, M13, tobacco mosaic virus, bacteriophage T2, and this is a chlamydia is a bacteria, and this is E. coli. So we can see the viruses can be very small to large in size, as large as chlamydia, but they are still much smaller than a bacterial cell like E. coli. So genomes, as I already told, it can be a DNA or a RNA. The RNA viruses are most of the infectious viruses which infect human beings are all RNA viruses. Most of them. There are few DNA viruses also, but majority of them are RNA viruses. And RNA viruses, actually the RNA genome is more primitive to DNA genome. So DNA viruses have evolved later than the RNA viruses. So they are single-stranded or double-stranded. They can be linear, circular, or segmented in configuration. There are some viruses in which if they have 11 genes or 10 genes, they have 10 different segments of genome. It's not chromosome, but it is separated. And in the viruses which have segmented genome, like influenza, like rotavirus, there is more higher probability of recombination events, and therefore they have lot of genotypes and lot of serotypes of these viruses, because what happens is, since when they form virions, if there is a co-infection of two different genotypes, then what happens is there is a high probability that some of the segments of one serotype and some of the segments from other serotype will combine when the virus is configured. So that is why in case of swine flu, when we had the pandemic H1N1 infection, pandemic in 2009, we found that that particular virus had segments from swine as well as from human, and the reason was only due to this recombination which happened in the swine. And the single-stranded virus genomes can be positive sense or the negative sense. Genome size ranges from 3,500 nucleotides to 235 kilobytes. The physical nature of nucleic acid dictates the strategy of replication and forms the basis of classification. See, when there is an RNA virus, whenever it will infect the cells, the first thing it has to do is it does both. It will replicate its genome. RNA will form another RNA using its own RNA-dependent RNA polymerase. But then to translate its protein, it has to again convert from RNA to DNA to make a cDNA. And then again, DNA is converted to RNA for transcription. So parallelly, both the replication and transcription occurs when there is a RNA genome. So this is how the classification was done, and In case of RNA genome, it can function as a mRNA or it can act as a template for mRNA. And in case of HIV, like a retrovirus, it genome is a template for DNA synthesis, like because they have a enzyme called reverse transcriptase, which will convert the RNA to DNA. Life cycle. So, since I said that they are the intracellular pathogens, they cannot replicate without the machinery and metabolism. So, therefore, they require host machinery, and the replication cycle therefore differs between the host as well as category of the viruses. But there is a basic steps which are required for every virus to infect any host cell. The first is attachment. The viral proteins of the capsid has to attach to the cell membrane with a specific receptor, and this actually causes the tropism. Like the influenza viruses, there are many genotypes. However, not all genotypes infect the human. They are like H5N1, bird flu. Bird flu has not attained the ability to infect human. Therefore, till now, we have not got a pandemic or an epidemic of H5N1 in human. It has this specificity only for the bird. So therefore, it can only infect the bird. So first is attachment, which actually is the main source of tropism in the viruses. Penetration, once it binds to the receptor, it will get inside the cell. It can be either to fusion to the cell membrane or it can be due to endocytosis. Anything. Uncoating. Once it is inside the cells, what it has to do is it has to first remove its outer capsid protein, outer membrane. So it removes the outer membrane and releases the nucleic acid. So the viral capsid is removed and degraded by either the viral enzyme or the host enzyme, and then the nucleic acid is released into cytoplasm. Some of the viruses they replicate in the nucleus. In that case, the nucleic acid gets inside the nucleus, and if they are the replicate in cytoplasm, it will replicate in the cytoplasm. So this will result into de novo synthesis of virus protein as well as genome. Once the virus has replicated its genome, what it has to do, it has to assemble. It has to assemble into progeny viruses, which we call virions. So then for this assembly, the genome is translocated outside into nucleus again, and it uses the endoplasmic reticulum and Golgi apparatus of the host cell to do the final assembly, and then it is exited out the cell. The exit is also of two types. One is lytic, one is budding. Lytic means when the progeny will go out and it will kill the cell. The cell will burst and the progeny will come out. And the second is budding, in which the cell is not killed and they just fuse through the membrane, and then by exocytosis they are budded out. So there are two types of virion release. So now, as we know what viruses are and how the viruses infect the cells and how they replicate, we would now go into what we can do or how can we control the viral infection. So for that, some of the common aims of the medical research are: first is to understanding the health, prevention of disease, diagnosis of disease, reducing transmission in case of infectious disease, developing therapeutics, evaluating treatments and procedures, vital statistics and trends. So these are the common aim which is required for any medical research, whether it is for bacteria or virus or fungi. However, not all of them will can be done by non-medical fraternity. Like understanding health, we need a medical fraternity who will do this, and also evaluation of treatment and procedures like doing clinical trials. That also we need a clinical team, and also vital statistics and trends. We need an epidemiologists and statisticians who would actually do this research. So, as a biologist or a non-medical scientist, how we can do a research which can actually aid or may aid us to find some solutions towards control of diseases. So first I would go about viral diagnostics. The importance of diagnosis is that unless we diagnose, we do not know what we are treating. Like if we have a viral infection and we keep on giving antibiotics, it actually makes the patient worse. What happens is it takes the body's immune system gets dampened, and that is why you are sick for longer period of time, and also you develop the antibiotic resistance. And also diagnosis helps in prevention of transmission, because once you know that you are not that this person has a viral infection like influenza or dengue, you can take proper precautions so that it is not transmitted to the other people. So the question we always ask is that why do we need diagnosis when no antivirals are available? The reason is because the clinical symptoms are overlapping. Symptomatic treatment may complicate the symptoms. We also need to, as I already told, that if we keep on giving antibiotics for a viral infection, then what happens is it leads to the increasing AMR. Plus, it also leads to longer. It takes longer recovery for the patient because of the side effects of the antibiotic. High epidemic pandemic potential with the huge mortality rates. It is important to monitor and provide necessary support to contain the outbreaks of infectious viruses, and also for isolation and advocacy for proper precautionary measures. Because supposing if we take the example of COVID-19, if there was no early diagnosis, then the pandemic's outcome would have been much, much worse. Because as the scientists identified the virus and found that it has a potential to spread across, immediately isolation, use of masks, etc. was recommended by WHO. So, the common symptoms, as I said, if you look at the respiratory infection symptoms, whether it is caused by influenza or it is caused by a uh, bacterial pathogen which causes pneumonia, they are all very similar. Like you have body aches, fever, difficulty in breathing, low oxygen, blood level loss. It can also lead to loss of consciousness. So there are different types of diagnostic tests. They can be broadly classified into detection of living pathogen, detection of antigen or antibody, detection of pathogen nucleic acid, and the desirable features of all diagnostic tests are high specificity and sensitivity. Specificity means that the test will only detect the virus it intends to detect. If the specificity is low, then it may give a false result. Supposingly, we are going to test for a influenza virus, or we are going to test for an HIV, but the test is not very specific. So it may detect any other pathogen and give the test as influenza or HIV, or it can miss, even though the patient has positive, but it will give negative. So then that is called sensitivity. But specificity is that it will only detect the particular virus which you want to detect, not any other. Quick turnaround time means the test results should be within few hours so that the clinician can provide the treatment on time. Ability to multiplex. If we can detect multiple viruses or multiple pathogens in one test, that is much better for a clinical outcome, because as I said earlier that for the respiratory infections the symptoms are very common. So it is the person who's suffering from RSV or influenza or COVID-19 will have the same symptoms. So if a person has to test multiple times for detection of that, it will take longer time. But if we can put all the tests in single clinical sample, the results we can get is within the same day. So these are the examples of the molecular test by the complexity level. Like the cost also goes up with the complexity. So we start with the basic serological test, go up to PCR, RT-PCR, for quantitative RT-PCR, that is real-time RT-PCR to know the pathogenic load. Multiplex PCR when multiple targets are at one go. High throughput is when these tests are commercialized and by in a single sample they can detect 16 or 20 pathogens. And the highest one is next gen sequencing or novel pathogen identification in case where no tests are available or if we do not know what is the cause of an outbreak, then next gen sequencing can provide the results. But the cost also goes up as we go to the complexity. So the traditional techniques for virus culture, we can, if you infect the clinical the cell lines with the clinical sample, you can see cytopathic effect after 24 to 48 or 72 hours, depending on the virus. This was considered a gold standard once upon a time. However, now its utility has gone down. It detects a viable pathogen, and the advantage is that you can have availability of live virus strains, which are used for not only vaccine development but also testing of antivirals and other things. The disadvantage is that it takes longer time. It may take days to weeks. It is laborious for detection of multiple infections, and this is how the uninfected cells look. And after infection with the virus, the cells would all turn round and die, show cytopathic effects. Electron microscopy. It is the only method to identify and detect unknown or new viruses. There's no antibody for which no antibody or sequence is known. The novel viruses such as Ebola, SARS-CoV, etc., were first identified and confirmed by EM. The major limitation is that instrument is very costly and the specimen preparation is also costly and it needs a technical expertise. If you look at these pictures, you can see that the viruses look different and based on their appearance and structure, the electron microscopists can determine what type of viruses may infect. These are the EM of different emerging viruses. We look at the if you just look at the structure, and each one looks very different. If you look at the Ebola virus, it looks like a thread-like structure compared to other viruses which are like COVID-19 and all, which are like round, sun-like structure. And this one is dengue virus. So the next is the rapid virus detection method. These are all antigen antibody based tests. These are rapid. They have advantage of using in the field if they in case of outbreak, but then these are less sensitive. That is a disadvantage of these tests. In this, we just take a clinical sample. Like over here, I am giving example of the rotavirus test. So we are taking the fecal sample and just inserting the swab into the sample collection tube, swirl it, and then use that solution to put a drop on the test kit. And then within 5 minutes, you can see the colored line, which will tell you whether the sample is negative or positive. The rapid detection kits are available for rotavirus, norovirus, as well as for influenza, SARS-CoV, dengue, NS1 is the most common one which is used now. So these have advantage of providing very rapid results, but the sensitivity is only around 60%. So there are chances that you may have a lot of false negatives. The next antigen antibody reaction which is used is ELISA. So there are two types, indirect ELISA and the sandwich ELISA, in which either we use the antigen coated well in case we want to check the IgM or IgG antibody in the patient, and then we will use an antigen coated well, and then we add the clinical sample, and then after binding we can add the enzyme linked secondary antibody, and the substrate is added, which will give a colored reaction. In case we want to detect the antigen, then we preferably use sandwich ELISA, in which the well has already a antibody coated against that particular virus. So we add the clinical sample which binds to this antibody, and then we wash, and then we add a second monoclonal antibody which will again bind to this viral antigen, and then we can add the substrate which is converted by the enzyme into colored protein, and this will give an idea about the sample is negative or positive. So the immunoassays have the advantage of being rapid, cost-effective, easy to perform, quantitative. Most of the clinical diagnostic labs, they still use ELISA as the frontline test for detection of many viruses. But there are a lot of disadvantages because they are not as sensitive. Uh, it depends on when the patient sample was collected, whether it was collected during early stages of infection or after 7 days of infection. The false positive or negative reaction may occur. And there is also cross reactivity among the same family of viruses like dengue and chikungunya. They may give a false cross reactivity. So therefore, even if we get dengue NS1 positive, usually it is further confirmed by a second test, like dengue IgM test or nucleic acid test. So the revolution in viral diagnostics actually came when the nucleic acid amplification tests were uh, invented. So these amplify the targeted regions of the viral RNA or DNA, and that is why they become very popular choice of diagnostics. They have a low turnaround time. There's a rapid onset of targeted treatment, labor friendly, superior sensitivity and specificity. You need very less sample volume. 100 microliter is enough. And you can also develop it into a high throughput system with multiplexing and automation. So this is the example of PCR, which is we call as xeroxing of DNA. And I think because most of you are uh, teachers in colleges, so you already have an idea about what is a PCR. So I won't go into detail, but there are just three steps: denaturation, annealing, and extinction. And there is an exponential amplification. The only important thing in diagnostics of a infectious disease is that whenever we are doing PCR, we have to have a known positive control always and a known negative control always in every test. The reason is because the PCR may not work as expected. So if your positive sample does not show result, you can deduce that the PCR reaction did not work. However, and if your negative samples give a band, then you can be sure that there was a cross contamination. Because when we are giving a result to a patient, we have to be very careful about false positive or false negative. So this is an example of how we run PCR with the clinical sample. These are different enteric viruses, rota, noro, adeno, and astro, and they run at a particular size. They give a particular size of fragment so that it can be differentiated using a positive control. This is a positive control. This is a DNA ladder. This is an example of the multiplex PCR. Over here, we can see that this is a detection limit from 10 to the power 10, 9 viruses per ml to 10 to the power 2 viruses per ml. And we can see that each of the particular virus has a different detection limit and the different size also. So we can actually test 4 viruses in a single reaction. This was same thing in a different in single plex, and then when we combine that, this is the same thing in a multiplex. So instead of running four different reactions, now we can run one reaction and we can detect all four viruses in one PCR reaction. So the time is saved, the reagent cost is saved. So for the RNA viruses, we have to add, first step is reverse transcriptase, because first we need to convert the RNA genome into DNA genome. So that for that we use a reverse transcriptase enzyme, which is purified from the HIV virus. So the first step is RT, which will convert the mRNA to a first strand cDNA using a random primers. Or in case of viruses, we do not use oligo DT primers because we do not have always poly A tail over there. And then after the cDNA synthesis, we do the regular PCR as we are doing it for DNA viruses. Quantitative real-time PCR, which is QRT-PCR. This is a quantifiable parameter. The advantage is that we can also know the viral load, whether the person has very high infection rate or has a low infection rate. In this, PCR is standard RT-PCR, but we just add a probe or a dye to generate fluorescent signal from the product. So as the PCR reaction and the DNA content increases exponentially, the fluorescence amount also increases exponentially, which is measured by the machine. So I'll just give this as example. There are two types of real-time PCRs. One is cyber-green one, and one is Tachman probe-based. Tachman probe-based real-time PCR is used for diagnostics. Cyber-green RT-PCR is usually used for research purpose. In cyber-green, we just add the fluorescent green dye in the PCR reaction. So whenever the DNA is amplified, this dye gets interconnected with the newly formed DNA. And as the amount of DNA increases, amount of fluorescence increases. So this is not very specific. Therefore, this is used only for research. But in case of viral diagnostics, what we do is we use a Tachman probe in which we have a primer 1 and primer 2, as in case of PCR. We just add another specific sequence which we call probe. And this probe has a reporter and a quencher at two ends. So when the probe is free, it will the quencher will inhibit the reporter. So there will be no signal. It quenches the signal by the reporter. But when the PCR reaction happens, as the Tat polymerase will go up, it will cleave because this probe is binding to the cDNA. So as it will go, it will cleave off this probe. A probe is degraded, and when the probe is degraded, the reporter is released. And once the reporter is released, it is away from the effect of quencher, so it starts giving the signal. And as more and more DNA is formed, more and more free reporter is released in the cellular reaction, and more signal is released. So this is how it is very specific. So not only the primer is specific to virus, the probe is also specific for particular same in the same gene segment for the virus, adding the specificity. This is how the reaction of the qPCR looks like. There is a lag phase and then there is an exponential phase, and the input is inversely proportional to the threshold size. So what we call is, this is a thing in which we call the city, where the lag phase starts converted into exponential phase. And based on the city value, we can determine the viral load. The higher the viral load, the lesser will be the city value. Say over here, the city value is 16 compared to 32. So that means this sample has a lower viral load compared to this sample. So this is the most popular choice of diagnostics. So then what can we next do to control the viral infections? I have already described the diagnosis and its importance, but that is mostly for identification and also to take preventive measures so that we do not transmit after knowing that this particular patient has a virus infection. But to control the viral infections, we have to either develop vaccines or the antivirals. Vaccines, there are available for few viruses. They are good, like it is available for influenza viruses. It is available now for SARS-CoV-2. It is available for rotavirus. They are good for reducing severity or death, but due to very high rate of mutations and large number of variants in RNA viruses, they do not prevent the disease. For example, SARS-CoV-2 vaccines or the influenza vaccine, it has reduced the mortality, it has reduced the number of deaths, but people still have got infected. They still get infected every year in spite of getting the vaccines. Antivirals. They are available for very, very few viruses as of now, like influenza, HIV, hepatitis C. These are the common viruses against which we have antivirals. These target viral proteins. However, viruses have also shown to develop resistance due to against these antivirals because due to selection pressure, the viruses develop mutation in that particular uh, protein, and then the antiviral does not work. The best example is Tamiflu. In the countries where the Tamiflu is used indiscriminately, now it is the influenza viruses are resistant to it. So, what are the future directions? We have to develop more vaccines using advanced platforms and also develop newer low cost antivirals to prevent the mortality. So, as I already told, there are vaccines and antiviral drugs. So, what are the types of viral vaccines we can have? We have four different types. The first is the most traditional was inactivated virus. When what people did is when they could grow the virus in a cell culture, they would inactivate the virus or heat kill the virus and then use that to inject the people or the animals to see whether the immunity is improved. That is the first stage of inactivated virus vaccines. These had disadvantage and advantage. The disadvantage was that you had to continuously grow the viruses all the time, purify them, and then also inactivate and inject. However, the immunity developed was not very lasting. The next came live attenuated virus. The live attenuated means the viruses are whole, they are weakened. They can induce the immune response, they also replicate, but they do not cause the disease. So they are more effective compared to inactivated viruses. Like for the influenza viruses, WHO identifies the strains every year, and then every year the vaccines are developed for to give it to people because those vaccines do not are not effective in another next couple of years. But in this case, also the cost is very high because you have to grow large amount of virus in the cell cultures, and also in culturing, you the viruses also develop mutations, and there is always a risk that the attenuated virus may become active inside the host cell if the host is immunocompromised. Then to bring the cost lower, the next development was subunit vaccine, in which the antigenic part of the virus, that is mainly the capsid part, is used, cloned into the adenoviral vectors, and then used to inject the host. These are less expensive because initially, once it is cloned, it it is very easy to grow them. You do not need the cell culture part to grow them. So that is why now subunit vaccines are more popular. In SARS-CoV-2 also we saw that Covishield was a subunit vaccine which had the capsid protein of the SARS-CoV-2. And the Covaxin was the live attenuated virus vaccine. DNA and mRNA vaccines. The DNA and mRNA vaccines were first seen or heard by most of the people was during COVID time because we had a mRNA vaccine which was given in USA and other developed countries, and the concept was that this mRNA once injected inside, it will synthesize the antigen inside the body. The same antigen which was used for subunit vaccine can be used for mRNA vaccine. It is safe and easy to produce. However, the cost is very high because mRNA is very unstable. So you need a cold chain like -20 temperature for transport. The development of vaccine. However, for developing a vaccine and bringing that to market usually takes 10 to 15 years. Because first we start in the laboratory doing an exploratory research in the cell line, then we go to mouse model, which is called preclinical studies. After getting successful results in these stages, then it goes to human clinical trials, which goes to phase 1 for safety and toxicity only, then phase 2 to see whether it is inducing the immunogenicity or not, and then to the phase 3, which is a much larger multi-site uh, clinical trial. After that, FDA or the CDSCO approval comes, and then the manufacturing happens, resulting in a longer traditional timeline, making it very difficult for countries like us to actually develop or invent vaccines from this stage. However, there are many vaccine companies in India which are actually doing from this stage, like Serum Institute, Bharat Biotech. So all the vaccines which are developed abroad and have passed the clinical trials for the cheap manufacturing, they are manufactured in India. But the COVID was a different thing. During the COVID pandemic, it was the first time in the history of mankind that 11 vaccines were granted emergency use by WHO within 1 year. So these are just the example. Like we have Covovax, Moderna, Johnson's and Johnson's, and Covaxin and Covishield, etc., which were all approved within 2021 and also injected to people. And it is the first time that this happened due to the emergency situation. So we have Covishield over here, which was licensed in 15 February 2021. This is a non-replicating viral vector. So this is a, as I told, subunit vaccine. And this is inactivated Bharat Biotech Covaxin over here, which is a inactivated. So these are the COVID-19 vaccines which are used in India, Covishield and Covaxin. One is adenovirus-based DNA vaccine, which encodes only the spike protein. This is a subunit approach. 2 doses, efficacy was 74%. Covaxin was inactivated SARS-CoV-2 whole virus, and its efficacy was 81%. The next is development of viral therapeutics. So over here, I am just giving example of rotavirus because my lab primarily works on rotavirus. We also do some research on influenza and SARS-CoV-2, but mostly on rotavirus. So there are actually, as I said, there are 3 different approaches towards controlling viruses: vaccines and antivirals. Among vaccines, which is we know that they are promising trends. They reduce mortality and hospitalization rate. They are prophylactic, so you can give it in advance to prevent the uh, outbreaks. But they are very population and region specific. Vaccine resistant escape mutants develop very fast. In the antivirals research, we have 2 types. One are the drugs which target the viral proteins. These are called VTAs, virus targeting antivirals. These are virus specific, and they are drug targeting host determinants, which are host targeting antivirals. Why do host targeting? Because the viruses require host to grow or infect. Otherwise, they cannot infect. And the second is there is a tropism. So viruses are host specific. It's not that all the viruses infect all the hosts. So therefore, host targeted antivirals is also one of the antiviral drug development aspect. Then in addition, the hosts are non-mutable targets. Like we do not have that high mutation rate because we have a proofreading ability in our replication. And there's also it can be less cytotoxicity if we can use a drugs which can be either herbal or ethnomedicine. However, the combinatorial approach is the best, like vaccines as well as antivirals to control any virus. So these are, as I already told about vaccines earlier, so I would just say there are two types of antivirals, like as I said, viral factors as well as host factors. Viral factors, antiviral drugs are either synthetic or it can be herbal or it can be drug repurposing in case of host factor. The majority of antiviral drugs that have been approved by FDA as of now are usually directly targeting the viral imported factors. The host factors are still in uh, development stage. So how do antivirals work? The ones which are targeting viral proteins, they either target the nucleic acid polymerases, proteases, integrases, or the neuraminases. So depending on which stage they are acting. So over here, if you look at the example of SARS-CoV-2, we can see that the drug can either impact the entry of the virus by binding of the virus to the ACE2 receptor, or it can inhibit the release of RNA release, the nucleic acid release into the cell, or it can inhibit the replication of the viral genome. Similarly, of this remdesivir, which is also used in case of influenza, it also binds with the RNA-dependent RNA polymerase, so it inhibits the viral RNA synthesis. This is how the antivirals which are developed against the viral function work. Most of the viruses I told have mainly encode only polymerase, either DNA-dependent DNA polymerase or RNA-dependent RNA polymerase of their own. So therefore, these are the enzymes which are target of most of the antivirals. In case of host, we can target chaperone proteins, transcription factors, cell cycle regulators, etc., which the virus requires during its replication. So, antiviral drug resistance is develops against the viruses when you target the drug against the viral proteins. Supposingly, we have HSV virus in which the drug targets the viral thymidine kinase. Similarly, varicella zoster, which causes the chickenpox, same the drug targets the viral thymidine kinase. For flu, it is viral DNA polymerase. And what has happened is viruses during their replication, they have developed mutations due to selection pressure so that these particular drugs do not work anymore on these particular enzymes in the viruses. So this resistance develops when the viral replication is not maximally suppressed. When there are escape mutants, then what happens is escape mutants have evolutionary significance, so they grow much more. This is an example. We have antivirals. This is Tamiflu or Seltamavir. And these strains, H1N1 pandemic, they have already developed resistance to by developing this mutation, H275Y. Similarly, Peramivir, Zenamivir, these also have developed mutations by H275Y, R294K, etc., and leading to the resistance in the by single point mutation. These viruses have developed resistance against these antivirals. So therefore, now the newer concept is to target the host determinants because the host determinants will not get mutated so fast because they are also part required for the host uh, maintenance of host genome. So I'll first show this. The host factors are evolutionarily conserved. So targeting host factors which help in virus replication. It will be harder for viruses to develop resistance. And it is also an interesting approach for development of pan antiviral. The reason is because most of the RNA viruses, when they infect the host cell, they actually utilize many of the pathways which are common among these. So if we develop a antiviral against a specific pathway, it can be used as a pan antiviral during the outbreaks. So for this to understand which of the host proteins and or the host factors are important for the virus, we can use the omics to identify that. Omics can be genomics, proteomics, or the metabolomics, which will identify the changes in compare the differences on the proteome or the RNA, mRNA sequences among the cells which are virus infected or controlled. So the proteins which are modified only after virus infection, they can be used to as a targets for testing whether they have a proviral role or they have an antiviral role. And once we know that this particular XY protein is has a function as a proviral protein, we can develop antagonist small molecules against it to develop the antivirus. So the first way we can develop antivirus, which is much faster against the known or the host protein which we have identified as the crucial for virus infection, can be used of traditional medicines or the herbal medicines. So, by definition, the traditional medicines include homeopathy and Siddha and Ayurveda, etc. And they use not only plants but also derivatives of natural products to treat the disease and enhance the general health and well-being. So there is a history in India and China and Egypt as well as Greece, Central Asia for the use of medicinal plants for various infectious diseases. So if you look at why we can use the traditional medicine over synthetic antiviral drugs is because we can it can impede the viral replication without affecting the host physiology or has a limited side effects. It may exhibit potentiality to modulate the immune responses of the host against viral infections, and it is affordable and easily available, and we can actually bring it to market much faster because of the because they are already used for many centuries in the country. So these are the antivirals which are already used or which have been developed from the medicinal plants against influenza, HSV, HIV, dengue, GIV, etc. So what is the mechanism of action? Mechanism of action is either inhibition of viral entry, that it can block viruses binding to the host cell. It can be inhibition of virus replication, targeting the host factors which are utilized by the viruses. Immune modulation, it enhances the body's immune response, activation of interferon, programmed cell death, and the anti-inflammatory effect. So if we can reduce the tissue damage caused by the viral infection, we can actually cause less morbidity and the patient recovery is much better. So this is just a methodology to study antiviral property, which I am not going to explain right now. So, so as I said that in our lab we are working on RNA viruses, rotavirus, which belongs to Reoviridae family. So I will just show you a little bit of research data from the lab. It is actually a major cause of childhood diarrhea. It has a wheel-like appearance. That is why it was called rotavirus, and it was discovered only in 1973. So the route of transmission is fecal oral transmission, and the difference between another advantage of most of the viral infections is they are democratic. You have viral infections both in the developed countries as well as the developing countries, including rotavirus. unlike bacteria, which are more common in the developing countries like us. So this is the data from 2017 to 2024, and we can see that out of 5,855 children who had come to the hospitals for with diarrhea, we had almost 2,700 which were positive for enteric viruses, including rotavirus, adenovirus, estrovirus, and norovirus, virus, among which rotavirus is the most common virus. The vaccine was introduced in West Bengal in 2019. In India, it was introduced in 2016, but West Bengal was got it in the last phase in 2009. So this is the same data in a different form. We can see that how the samples have, how the viruses are differentiated between the from 24% rotavirus to 0.4%. Over here, this is like if we consider this as 100%, then we see rotavirus as 51% of this 47%. But this is 24% is of the total 5,855. So please do not get confused. So see, if we look at the seasonality, we can see that predominantly winter months we get more infection with and the norovirus we see more or less all through the year with not too much of change, but rotavirus predominantly peaks are in the winter. So we looked at we had one observation that the rotavirus NSP1, which is one of the protein encoded by rotavirus, activated NF-kappa to inhibit the premature apoptosis, which was caused by rotavirus in 2010. And then to see that whether we can inhibit NF-kappa B and whether that will have a better impact as an antiviral, we screened the various phytochemicals which have inhibitory effect on NF-kappa pathway, starting from gallic acid to epigenin, quercetin. These are all phytochemicals isolated from various plant extracts. And we found that the quercetin was the best among all the phytochemicals in as an anti-rotaviral because its IC50 was only 4.36 micromolar, and it also inhibited various rotavirus strains starting from human to porcine to um, equine. So it did not had any host specificity. And over here, if we look at these are the normal cells in which this is red color means infected rotaviral protein and blue is nucleus. So over here we can see the replication is over here. This is only 6.25 micromolar, and over here as we increase the dose of quercetin, we can see there's less and less number of infected cells over here. So if we use 50 micromolar or 25 micromolar, we almost see inhibition of rotavirus replication. There is not too much effect on at 6.2 or 12.5 micromolar dose. So then we did whether in the cell line it may have an effect, but what about in the animal model? So that is why we also checked in the animal model to see what happens. And over here, if you look at the data, we can see that in the pups, we infected the pups with the rotavirus and also treated half of them with quercetin and half with only water. And if we look at the infection, we can see that at 48 hours, we can see expression of the rotavirus protein in absence of quercetin, but in presence of quercetin, there was much less expression. This was the viral plaque assay in which we measure the uh, virus particles in the intestinal wash as well as the stools which we collect, and we can see there is much lower numbers of viral particles in presence of quercetin. This slide shows H&E staining of the intestine These are the control intestine where we can see the villi is healthy and long. These are the infected cells where you can see the villi are ablated. They are shortened, they are disrupted. This is the 40x. You can see how the villi has been ablated in case of virus infected mice. But in case you have this infected mice and in addition you treat with quercetin, you can see restoration of the villi structure. So this suggested that quercetin has a good antirotaviral potential. Similarly, the next one is that HO1 axis. HO1 axis is the heme oxygenase 1 enzyme, which is a cytoprotective gene and which is regulated by the transcription factor NRF. We observed that after rotavirus infection, the HO1 was actually degraded. It was downregulated. So therefore, to induce it, we used a andrographolite extract which upregulates HO1 in the cells. So once we induce HO1 in the cells, in addition to rotavirus infection, we can see that the rotavirus infection goes down. There is a much 50% reduction in the number of rotavirus particles in the cells after treatment of um, endographene. So in this case, HO1 is actually antiviral gene which is downgraded by rotavirus during infection. If we can upregulate that gene during the infection process by using any small molecule drug or a herbal compound, we can actually have a naturally antiviral effect. So these are just the example of how phytochemicals can be used for developing antiviral drugs. But there are challenges with the traditional medicines because of batch to batch variability. Due to natural variability of the raw material in different seasons, soils, manufacturing variations, there's very few pharmacokinetics studies in this because most of the time we study, we use only single compound isolated from an extract, but then this differs from a full extract. So these pharmacokinetic studies are challenging because of their chemical complexity. Many bioactive compounds in extracts are water soluble, but they are poorly absorbed due to their large size and inability to cross membranes. So there are risks when we combine this with conventional medicines without proper clinical trials. So there are lack of clinical trials in case of traditional medicine. So there is a requirement for scientific validation. There is also a requirement for regulatory approvals, and also proper identification and correct administration is required to avoid any toxic effects. So globally now there is a resurgence in demand to study and use traditional medicines. Pharmaceutical companies are also isolating bioactive compounds for developing new antivirals. The Tamiflu drug which we use for influenza was actually derived from star anise. There is plans for large-scale clinical trials and developing standardized extracts for the consistent effects. So we can say that traditional medicines have a great potential as pathogens, and they can impede the pathogen replication without affecting the host physiology. What we have to do is we need to identify the active compound from the plants, and since it is not easy to purify them again and again directly from the plant, synthetic molecules which mimic the phytochemicals can be synthesized because that would reduce the cost also and will have a consistent production. A multidisciplinary approach can lead to safer, effective treatment. WHO is also working and promoting towards integration of traditional medicines into uh, the health system in agreement with the Government of India to establish a WHO Global Center for Traditional Medicine in India. So as I said, told about the traditional medicines, we can also use synthetic small molecule inhibitors as host-targeted antivirals. Like I said, the omics can identify host factors. So we have done a mass spectroscopy to identify the various proteins or the changes in the proteome during rotavirus infection. And we found that these particular genes like ETM, CHK2, NF-kappa B, I already told, importin and NRF2, UPF1, etc., they were altered during infection. So I will first show about the DNA damage response. If you look at the DNA damage response proteins, we can see that in case of rotavirus infection, we see there is a increase in the phosphorylation of ATM. There was no change in the ATM expression, but phospho ATM increased, phospho CHK2 protein also increased. This is a protein, this is a marker for the rotavirus infection, BP6. However, this ATM CHK pathway normally is activated in absence of cellular damage, but in case is usually activated when there is a real cellular damage. But in case of rotavirus infection, we found there was no cellular damage because if you have a cellular damage, like in case of doxorubicin, you can see this comet-like structure, head and tail, but we do not see that during rotavirus infection. So this pathway was activated without, in absence of the real cellular damage. So when we looked at, we did the confocal microscopy, we saw that ATM and CHK2 were located in the viroplasms during the virus replication. Viroplasms are a dynamic replication factory inside the cells in which the virus replication occurs. So if you look at this early time point, you can see these small replication factories are around the nucleus, green, small dot-like green spots. Then as time increases, you can see there is an increase in size and the numbers of these viral structures over here. They look like this in the cytoplasm. Inside this, the virus replication occurs. And over here, we saw that both CHK2 as well as ATM, they were actually localized within these viroplasm during the replication. The question was that both ATM and CHK2 are DNA damage response proteins, so they should be present in the nucleus. However, viroplasms are in the cytoplasm, so what are they doing in the cytoplasm? So then to know their function, we use specific inhibitors for ATM and ATR, and when we use the inhibitor, we found that the number of viroplasms and the size of the viroplasm decreased drastically. This is the control, and this is in treatment with ATM inhibitor and ATR inhibitor. You can see there is very less fusion of viroplasm, and the viroplasms do not mature. So, in conclusion, we have said that the ATM and CHK inhibitors have antiviral rotavirus potential because they do not allow the maturation of viroplasm for the virus replication. So similarly, next we focus on importin B1 and RAN. This regulates the nuclear import of cargo. Over here also we can see that viral replication can regulate the access of host transcription factors into nucleus through the nuclear import path. So importin beta 1 together with importin alpha forms a ternary complex with the cargo molecules carrying the classical nuclear localization signal NLS. And this stable complex transports through the nucleopore into the nucleus, where the RAN GTP interacts with importin beta 1 and induces a conformational change, leading to the release of the cargo molecule. So if you look at the rotavirus infection, we can see there is a change in the nuclear import machinery. There is an induction of importin beta 1, induction of RAN during rotavirus infection compared to the mock infection. And this importin beta 1 facilitates rotavirus infection because if we use a siRNA, we can see there is a down regulation. This is in absence of siRNA, this is in presence of siRNA, we can see there is down regulation of rotavirus replication. This is same in presence of siRNA. You can see much less virus particles. So for establishment of protective replication, we need this nuclear transport machinery to be active. So both importazole and ivermectin, both are the ones which actually block the function of important beta 1. And once we use that, we can see they prevent the rotavirus infection. Importazole is a drug which inhibits importal beta complex. And if we use this, we can see that there is a reduction in rotavirus virus replication by plaque acid. If we use ivermectin blocks importin alpha subunit, not the importin beta. And over here also we see that there is a reduction of rotavirus particles or replication in presence of ivermectin. The SI index of ivermectin was more than 10 across RV strains, suggesting that it has a good therapeutic potential. I do not know whether you remember or not, but ivermectin was also used during SARS-CoV-2 pandemic uh, for treatment as an adjunct therapy. So when we estimated cytotoxicity of importazole and ivermectin in mice, we can see that ivermectin at the dose of 5 milligram per kg body weight showed toxicity in mice, but it was okay at 1 milligram or 3 milligram. There was not much reduction in body weight. Importazole we could use at 1 milligram or 3 milligram per kg body weight. At 5 milligram body weight, even importazole had 40% reduction in body weight. So we have used only 1 milligram and 3 milligram, 1 milligram of ivermectin and 3 milligram per kg of importazole in case of to study their antiviral effects. For ivermectin, we can see that at 1 milligram per kg, this is rotavirus infection plus drug. This is rotavirus infection with importazole. This is with ivermectin, and this is only rotavirus infection. So we can see the body weight of the mice have reduced drastically at day 7 in case of virus infection, but there was no change in body weight. We look at the virus particles, we can see that there were much higher virus particles in case of DMSO, but the viral particle number was significantly low. In this is in log scale in both ivermectin and importazole. Similarly, if you look at the small intestine at the day 7, we can see this is virus infected intestine where the villi is disrupted. And however, in case in presence of ivermectin and importazole, we can see the villi is restored. This suggests that the suppression of classical nuclear import pathways by importazole and ivermectin can inhibit rotavirus replication effectively in animal models. So, with this, I would like to acknowledge the funding agencies like ICMR, DST SERV, and Okayama University for funding our research, and all my past and present PhD students, because all the work which has been done are actually done by them. We only act as a facilitator. So without their hard work, I would not have got any of the awards which I got. This I'm done. If you have any questions, you can ask.
Thank you so much, ma'am. Am I audible?
Yes, yes.
No. Thank you so much, ma'am, for the insightful and engaging presentation. We will now move to the question and answer segment. I invite the participants to share their questions in the chat box, and I will take them up one by one.
Hello. Am I audible?
I have a question from Dr. Chatterjee. Ma'am, why antiviral curative direct medicines are sank in the market, although vaccines are plenty? Why we are not able to develop medicines directly against the deadly virus like rabies, although vaccine is available?
As I told earlier, that most of the viral infections, if most of the viruses which are highly infective are the RNA viruses, and then there are a lot of variants also in RNA viruses because of the lack of proofreading in the genome. So the viruses develop mutations very fast, and therefore the antigens keep on changing. So to develop antivirals is a difficult task for that reason, because due to the escape mutants, the viruses will, have, they will kind of evade the antivirals, and then again the antivirals need to be modified, which is not that cost effective. First thing, and the second thing, in case of rabies especially, because the rabies disease, when it is caused, it is the onset is very rapid, but therefore, if you give the drug also, it will not be as effective compared to the prophylactic, in which you can give the vaccines to the patient as well as to the dogs earlier, like so that they do not become rabid. That is why the antivirals are less in the market. The only antivirals which are successfully used are and have been developed are for HIV, because it is a lifelong infection. See, the companies will also develop a drug only if there's a market. That is a major problem. They will not develop drugs and invest for something which will not have market. In rabies, the patient will die. The chances of surviving is very less if you do not take the vaccine. So HIV and hepatitis C. Hepatitis C is also a lifelong infection. Once you get infected, you will not get cured. So that is why the companies have invested, and there are antiviral drugs against hepatitis C and HIV, but there are very limited drugs against the acute infections.
Thank you so much, ma'am. We have another interesting question by Dr. Namrata Ray. She asks, when drugs targeting the host determinants used by virus are designed, are there chances of harming the host cell function? If yes, then how is it checked?
Yes, actually, this is a very pertinent question. The thing is that, see, when we are using or we are trying to design a drug against any of the host protein, the main aim is not to delete that host protein, or not to remove the function of that host protein. The target is identified in such a way that when a virus is infecting the cell, that particular protein expression is induced manifold from the basal level. So, like, if it is expressed at the level of, say, 1, the after the virus infection, the level goes to 5, 6, or 8. In at, so when you are targeting a drug, you are using a drug so that that level goes back to 1 or 2, not that it will become 0. So, the host will have the basal level what it requires, but that additional upregulation is stopped. That is one thing. That is the way is controlled. And the second is, most of these host targeted antivirals are being developed only against the acute infections, the infections which are for 7 to 10 days, like influenza, SARS-CoV-2, etc., dengue, which are not like hepatitis and HIV that you have to eat the medicine lifelong. So then, if it's an acute infection for someone, a complex multicellular organelle, the impact will not that much for if you are taking a drug for 4 or 5 days. So
Okay,
that
ma'am. Thank you so much.
Thank you, ma'am. We have another pertinent question. Individuals affected with chikungunya virus usually do suffer from severe joint pain as post-infection complications. This pain also relapse. Why does it happen?
pain in chikungunya is actually due to the host immune response, and since the viruses, they replicate, chikungunya viruses, they are shown to replicate in the joints area. So therefore, the host immune response is activated and it causes the damage. And that since the damage is quite severe, depending on the the load of the virus, depending on how the host immune system is reacting, the damage may be less or damage may be more. There are people who for whom the fingers and the joints have bent, like for maybe a year or two, they are not able to use their joints. But there are patients for whom it only lasts for 10-15 days or 1 month, and then they recover. So it is mostly our own immune system. If we have a huge cytokine storm, our pathogenesis will be high. So it is it is because of that is why they say that you have to have anti-inflammatory drugs during chikungunya infection, and also do a lot of exercise and physiotherapy to recover faster instead of.
Okay, thank you so much, ma'am. Actually, I am suffering from such kind of problem. That's why. Thank you so much.
Thank you so much for the parting questions. Thank you so much, ma'am. Uh, uh, Ma'am, thank you so much for an illuminating and deeply insightful lecture. On behalf of UGC, MMTC, University of Calcutta, and Maulana Azad College, I extend our sincere gratitude to Dr. Mamta Chawla Sarkar for sharing her extensive expertise and for enriching this morning session with her valuable perspectives grounded in the years of impactful research. Your explanations, especially in the complexities of the virus, host infections, open research, your explanation, you new dimensions for understanding our participants. I thank all the attendees for their active participation and thoughtful engagement. Your presence and questions added meaningful to the session and contributed to its success. With this, we conclude the morning session of today's program under the online refresher course of Bioscience, Bioengineering, and Nanotechnology. We look forward your continued participations in the upcoming sessions. We wish you all a productive day. Thank you.
I just want to know what is the background of the audience, because I need to send questions. So I need to know what is their background, just broad, like they are from which biological sciences or chemical sciences or what.
Madam, from chemical science, like I myself, I am from chemistry, but most of them are from biological background. So also there is physics. So there is pure science and biological science both.
Okay. Okay. So if they are
No
pure
point.
science, then physics and all, then I have to keep the questions very basic and simple. So that is why I was asking.
Uh, Participants, can you please tell a brief about your background? Ma'am wants to know. If anyone is interested, please open the mic.
I just want to know how many are from biological sciences and how many are from other pure sciences. Like that's a percentage. So
So I am from biological sciences from Department of Biochemistry.
the biochemistry is okay. It's like kind of
Uh,
you were taught microbiology in biochemistry also, I think.
yes, yes.
Yes,
Both.
yes. So no,
I
I
had a
just
Uh,
question if you have time.
uh, Thank you, ma'am. And our next session is starting very
okay.
soon.
Okay. Thank you.
Okay.
Thank you.
Thank you so much, ma'am. Thank you so much.
A warm and a very good afternoon to one and all present over here. Our next speaker, Dr. Kutubuddin Molla, has already joined. Let me take the pride in to introduce Dr. Molla before you. I'm Dr. Dipan Adhikari, Associate Professor of Botany, Maulana Azad College. I'm taking the pride in to introduce Dr. Molla. I have been knowing him for a long time by his name, but today I got the rare opportunity to meet him and face to face. And they got last chance, a very opportune chance to listen his lecture. So Dr. Kutubuddin Mullah is a senior scientist in agricultural biotechnology at the ICR Central Rice Research Institute, CRRI, Cuttack, India. He earned his MSc and PhD from the University of Calcutta and completed postdoctoral research at Pennsylvania State University, USA as a Fulbright Fellow. Dr. Mullah's research focuses on precise genome editing in crop improvement employing CRISPR-Cas, base editing and prime editing technologies. His laboratory pioneered the development of a compact TNPB genome editor for plants as an alternative to Cas9, Cas12 system. These TNPB vectors are now used in leading laboratories across USA, China, Korea, Belgium, Canada, Argentina, and in more than 25 laboratories across India. The TNPB innovation has secured him a patent. His contributions to genome engineering and rice improvement has earned several prestigious recognitions, including the Jawaharlal Nehru Outstanding PhD Thesis Award, ICAR. Am I audible properly?
Yes,
Yeah,
yes, you
thank you.
are.
Thanks, thanks. I see here the INSA Medal for Young Scientist and Professor Shushil Kumar Innovation About NBRI. He was recently selected as an INSA Associate Fellow in the year 2025. He was also named a next generation leader by Stanford University to help and frame future guidelines for responsible biotechnology at the Spirit of ILOMAR Summit. ACE-LOMAR Summit. He is a member of the Indian National Young Academy of Science, INYAS, and has co-edited three popular volumes of the Springer series CRISPR-Cas methods. With this illustrious bibliography, I would like to hand over this mic to Professor Kutubuddin Molla for his next talk. We are eagerly waiting for him. Over to Dr. Molla.
Thank you very much, sir, for your kind introduction and invitation to this refresher course. And I think this is the last day of your refresher course, and I have a job to make you understand what is genome editing and how this genome editing can be applied, yeah, for biology, agriculture, and medicine. So can anybody tell me if the slide is visible in full screen mode?
Yes, sir, it is visible.
Yeah, great. Thank you very much. So, okay. So because I already learned from the principal that this refresher course has participant from biological science, from chemistry, and also one participant from physics. So I made my presentation in such a way that you would be able to follow this presentation, even if you're from physics, but you have to listen to them. Okay, that's the only thing that you have to do. So genome editing is a tremendous potential technology, and that's why, you know, after the first paper came on this technology, within 8 years, this technology has been given a Nobel Prize. And Jennifer Doudna and Emmanuel Charpentier has got Nobel Prize in 2020 in chemistry. So I'll be telling you how it is impacting the whole, modern biology, agriculture, and medicine. So let me tell you a brief outline of this today's presentation that I'm going to cover. And the first part, I will be talking about how a gene could be made non-functional, means the DNA could be disrupted so that it does not work in proper way. That's called gene knockout. And in part two, I'll be telling you how it could be used in, this. There are a lot of derived technologies. There's original CRISPR-Cas9 technology, and that gave rise to a lot of technology that can also alter, different kind of genetic expression like enhance or reduce or even modified different way. And in part three, I'd be talking about precise editing. What do I mean by precise editing? Yes, genome editing is a precise technology, but this precise editing, what I would be talking about is a more advanced version of editing, or you can change one single letter to another letter, especially as you want. For example, A to G, for example, C to T, exactly one to another rewriting application you could do. And in part for the last part, I would be giving you some wonderful, exceptional example of application across agriculture and therapeutics. The genetic disease could be cured with this technology. So let me start from the beginning. Obviously, biology people know it, but as to give you a refreshing of your knowledge. So human body are made up of trillion of cells, and similarly, any plant, for example, here I have shown a peanut plant. Uh, that is also made up of trillion of cells. And if I want to give you an example of a plant cell, how it looks like, this is a plant cell, and it has this nucleus. Okay, so let me go to this. Yeah, it has this nucleus, which is the head of the cell, because it contains chromosome in it, and this chromosome actually nothing but a long stretch of DNA that coil around some protein called histone protein. But just remember that chromosome is made up of long stretch of DNA, very, very long stretch of DNA. Now, the whole long stretch of DNA are not equally functional. Some part of the DNA are functional, and that is called, this is highlighted here in green color, is called gene. Now, this gene, why it is functional? Because it produces, after expression, it produces some protein. First, gene produces mRNA, and then it produces a protein. And this protein can directly or indirectly confer or control a character of any organism, be it a plant, be it a human, or any other animals. Now, what is there in our gene? If you see, it has only four letters in it, A, G, C, or T. These four kind of letters are actually the composition of DNA or gene. But the sequence of these letters are very, very important, and they make up the genetic code of an organism. And that ultimately controls how an organism should look like, how an organism should behave, and how an organism should respond to the environment. Now, let me go to genetic variation. Now, what is genetic variation? As I told you, that genome is made up of chromosome, and chromosome is made up of DNA, and DNA sequence is nothing but the sequence letter A, G, C, T. There are no other letters. Now, if you see here, this is the original sequence, but in some cases, you could see that this A has been modified, like is changed to a G nucleotide, and this is called a new genetic variation. Similarly, for example, in another organism, in another case, this G could be modified to a T nucleotide. That is also called a genetic variation. Now, there are other kind of genetic variation. For example, if it is the original sequence, and maybe there are extra, after this G, there is insertion of this GTA, 3 nucleotide or 3 DNA later is inserted. That's called insertion. Similarly, there could be deletion. 2 nucleotide is deleted. Like between T and A, this GC is deleted. So this is called deletion. So these are different kind of genetic variation that I want you to understand. Now, why this genetic variation is very, very important in case of evolution, in case of crop improvement, in case of trait improvement. For example, you can see here, the middle plant is the original plant. That's an ancestor plant called Brassica oleracea, and this plant has gave rise to all the vegetables that we eat today. So from a single plant, it gave rise to kohlrabi, cabbage, cauliflower, broccoli, kale, all those vegetables that we eat today. And now, how this single plant has developed to different kind of vegetables, it's not natural, it's not entirely human-made. So it's a combination of nature and human. How? Because in nature, when this plant grows, then it acquires some kind of genetic variation, which is actually generated because of a spontaneous or natural mutation. Spontaneous or natural mutation means nobody is doing that. In nature, Sometimes the gene sequence or DNA later get changed over time, and that causes genetic variation, as I just showed you in the earlier slide. Now, if this genetic variation could generate a new trait means new look to the plant or a new feature to the plant or new feature to the organism. And it may give rise to a new trait, it may not give rise to a new trait. Okay. Now remember, if genetic variation gives rise to a new trait, and if this new trait is beneficial to human, then humans select them artificially. And it means when humans select them artificially, and it means they are actually indirectly selecting the genetic variation that gives rise to the new trait. So it's a spontaneous mutation generated in nature. That's a natural thing. But the trait is getting selected by humans. So it's a combination of natural origin and then selection by human. And that gives rise to the development of ultimately this kind of vegetables from the same old plant. Okay. For example, if I want to make it very simple, for example, you are growing this plant for a long time. Maybe your forefather is growing this for a long time. But this portion of the plant are very, very liked by the daughter of maybe our forefather. Now, now, for example, if once in a morning, like after a few years of cultivation, now our forefather sees that one of the plant has a bulky or a swollen stem here, and he will be immediately very happy because a little more amount of vegetable would he would be able to give to his daughter because this plant has a thicker stem. Now, because this is beneficial to that farmer or to that forefather that I talked about, now you will select or isolate that plant separately that this is the most interesting plant, and you will collect seed from that plant, and you will grow that for the next generation. And you will see that okay, most of the plants will have the swollen stem. So that's the one generation of selection. That's one generation of selection. Okay. Now, for several generation of this kind of selection, ultimately gave rise to kohlrabi. Now, if you ask me how this plant once fine a morning actually gave rise to swollen stem, it is because of a spontaneous mutation or spontaneous change in the DNA, and that gave rise to swollen stem, and this swollen stem is selected by the human. This is how we domesticated our crop. Wild crop became cultivated crop because of this kind of situation. Now, up until 20th century, this kind of spontaneous mutations are the only source by which the trait was developed, and those traits are selected by human. Okay, but after 20th century, human has learned how to change, and human has already wondered how to rapidly make genetic variation or modify changes in DNA. So what they have done, this is one example to show you, just to give you an example. Here is a gamma garden where plants are being treated with gamma rays, which is radioactive ray. And once you treat your plant and the plant DNA, radioactive ray is very, very damaging to the DNA, and it can cause a lot of changes in all over the DNA. And after the treatment of the plant, you can just collect the seed and grow to the next generation, and you can get a lot of different type of traits, a lot of different type of variations in the phenotype, like how it looks like tall plant, dwarf plant, broad leaf, small leaf, larger flower, smaller flower, larger seed, smaller seed, a lot of different type of variations. But the farmer will next select which one would be interesting to them, and they will select it and grow for the next generation to develop to a variety. But remember, in this procedure, it causes a random genetic variation. It means all over the chromosome there will be changes. This all changes or variation. But one of the variation could be very, very beneficial, and that could generate the desired trait, and you would only select for that trait. But those undesired variation also exist in your selected crop, but they do not give you any output or outcome in the phenotype. So if I give you statistics, there are a lot of more than 3,000 plant varieties that we grow and even eat today are developed through this kind of random mutagenesis, or this is called mutation breeding. Okay. Now, If I want to share with you some kind of existing phenotypic variation, you see here a lot of rice plants, and these rice plants they look almost similar, dissimilar from each other. One is smaller, one is longer, cylinder. One is bold, smaller. Color is different. The same Oryza sativa, but they look so different from each other. Now, if I show you a same African cichlid fish, there are huge variation in the nature available. Here you can see dog. Okay, so these are all natural or created variation exist in the phenotype. Now, how new variation could be created? You can cross one variety and one breed of dog with another breed of dog, and you can make a genetic combination. You can give rise to a new kind of dog. This is how these are all variation that exist in nature. Now, my question is, can we mimic the natural genetic variation or generate novel genetic variation at a desired spot? What does it mean, desired spot? It means it should not be random, because we know which gene function in which way nowadays because of the modern biological advancement. Now can we mimic the natural genetic variation? It means, suppose this variation is existing in the nature, genetic variation, we know that this genetic variation is really, really beneficial for our farmers, for our people. Can we create that? But you can say that, okay, it is available in nature, but why don't you use that already available in nature? Suppose one wild crop has the genetic variation, but you cannot cross it with your cultivated one, and it's not easy to transfer that genetic variation to your to your desired crop. It's very difficult. This is one example. Second example is, suppose for example there is one plant discovered in China, and there is two DNA related difference, and you know that this two DNA related difference actually causes the crop or the rice plant grow in low nitrogen supply. Now, low nitrogen supply is a very attractive character because you can reduce the fertilizer input, and that would benefit us economically and also environmentally. But there is nil exchange between China and India. So you cannot get that germplasm here, and you cannot incorporate that genetic variation to our crop because China would not share it with you. Now, as you know from the literature that this genetic variation is beneficial, if we can generate or mimic that genetic variation in our crop, our farmer would be benefited. Okay, uh, that's the key I wanted to convey. Now, why you need to generate novel genetic variation? As I said, genetic variation is the key to crop improvement. If you can generate new genetic variation, you can generate a lot of new traits. So that is why you need to generate or mimic new genetic variation or mimic natural genetic variation. Now, how those genetic variation could be generated? By this modern technological genome editing, it can allow you to generate genetic variation or mimic genetic variation, and there are several tools. For example, here you can see JDFN, zinc finger nuclease, TALEN, and CRISPR-Cas9 are the first generation genome editing tool. And then next generation genome editing tools also come in the scenario 2020 onward. I will tell you later. So here is Dan Borders, who has actually pioneered the TALEN technology from University of Minnesota. CRISPR-Cas9, this Jennifer Doudna has got Nobel Prize in 2020 along with Emmanuel Charpentier. And there are some next generation genome editing tool I will tell you later. Now, if I want to give you a simple introduction to CRISPR-Cas9, the most popular one for which Jennifer Doudna got Nobel Prize, it has two components. One is a Cas9, which is a DNA cutting protein. Another is a small RNA, which is a customizable RNA. Now, if I want to give you a simple analogy, this Cas9 is a postman which has two scissors in its pocket. And now what you give to a postman, you give an address to the postman. Now, once you give the address to the postman, here is the address is the customizable small RNA is the genetic address, I call it. So once you give that genetic address to the postman takes it and it will go to the genome and it will find or search where is this address located. It means where is this sequence located? Because you have given the genetic address written as per your wish, okay? Now, once it finds a similar sequence, then it will bind there, and that is the signal to the postman, to the Cas9, to execute its job. So what is the job of this postman is to cut the DNA. With one scissor, one strand of DNA would be cut, another scissor, another strand of the DNA would be cut. Okay. Now, once this DNA is cut, it means that if it is a gene, then the gene would be non-functional. So this is a highly efficient tool to make precise uh, double-strand break at your target gene. Okay. Now, okay, it is understood that you can design a genetic address and it can go to your gene and make a double-strand break. But how the mutation or editing happens? Now, once the double-strand break happens, then this double-strand break happens in DNA, which is the genetic material. So it's a very serious damage to the organism. Now, once it is damaged, then cell will try to repair this two broken end, and by simply religating or resealing this broken end, while they will reseal, most frequent pathway is an HEJ, by which, while they will be resealing, there could be some error, and due to this error, some random insertion or deletion could happen. What does it mean? I told you insertion deletion already in my earlier slide. So random new letter inserted or original letter deleted. That could make the whole sequence of the DNA or the gene changed, or open reading frame is now modified. And as a result, this gene is no longer the same gene. So that's why this gene would not be able to function in the similar way as it was functioning in its original version. So this is how a gene could be disrupted with very high efficiency with this technology. Okay. Now, this video would make you understand how this actually works. Okay, so I think you got the idea how it actually work. Now, see, now if you ask me one question that, okay, what if I need to, I need to edit three different genes simultaneously, not a single gene, because there are a lot of traits for which multiple genes are involved, not a single called multi-gene trait or polygenic traits. So for example, in that cases also you can do multiplex genome editing, in which you need to only design three different addresses. For example, you want to target three genes. Okay, and you have to give that address to the same Cas9. For example, if I want to give you an analogy, suppose you want to send the same invitation letter to 5 of your friends. What you do, you write 5 different addresses, but the letter is same. Okay, so similar case here, and the same postman is giving that letter to 5 different addresses based on the address that you have written. Here also you design 3 guide RNA and you give them to the Cas9, and Cas9 takes them and go to their respective loci. For example, this address for gene 1, this address for gene 2, this address for gene 3. So all these 3 genes would be cut because you have made 3 different addresses in the form of guide RNA. And they will be repaired and they will be edited simultaneously. This is how multiple gene could be edited in a single go. Now, let me give you a simple example how it could be, how we have used this CRISPR-Cas9 technology in our application of crop improvement. What do you see here? Here, tall plant, okay, tall plant fall flat due to high wind and heavy rain. And climate change actually increases this kind of incidences. But during Green Revolution, we made our plant, like rice plant, shorter. So it's called semi-dwarf version. And this shorter version is more stronger. They do not fall flat due to heavy rain or high wind because they are stronger. Now, and that gave us the food security during the Green Revolution. Okay, all of you, this is a very good success of our food security application of crop input. Now, when people have analyzed why those plants became shorter or semi-dwarf in nature instead of taller, then they have found that this shorter plant, this is taller and this is shorter plant. The shorter plant has the same gene. The gene is present here, the gene is present here. This gene name is semi-dwarf 1 gene, but this gene is functional here, but it is non-functional here. So it means when the gene is non-functional, then the height becomes reduced. And why it became non-functional? Because it has a natural 383 base pair deletion. What does it mean? It means from that gene, 383 DNA later is deleted, and that was natural. And though that is a natural genetic variation that was used during our green revolution. Now, what we wanted to do in our laboratory, we want to mimic this green revolution allele in some aromatic short grain rice, which has never been made semi-dwarf, because when breeder has tried to cross, the semi-dwarf with this tall aromatic rice, and then they wanted to transfer this 383 base pair deletion genetic variation to the aromatic rice. But that was not possible because whenever the plant is becoming shorter, the aromatic rice becoming shorter, that's okay, but their grain shape become changed, their aroma is gone, so no farmers is liking that. Now if I ask you why it is happening, this is called linkage drag. So when you try to transfer one genetic variation by simply crossing, so while transferring desired gene, there will be some undesired gene also getting transferred, and that is because tight linkage of good gene with bad gene. And because the bad gene gets transferred, then the aroma is gone, then the rice grain shape is also gone. And as a result, the breeding was unsuccessful. Now, because of this tool, now what you can do, you can precisely only change or mimic that natural variation by making cut. Here, in a guide RNA genetic address here, another genetic address here we have designed, and then in between the region is 620 nucleotide or 620 later. Now what we have now generated plant which has 620, exactly 620 base pair deleted from the intervening region. So the DNA, no other portion of or no other genes are affected, only this SD1 gene was precisely targeted to delete 620 nucleotide very, very precisely, and now plant become dwarf because it does, and it has the intact grain quality because it does not impact any other portion of the gene and it does not involve any crossing. It's only modifying the specific target gene. So if you see the phenotype here, how it looks like after the 620 base pair deletion, you can see here this is the wild type or the original one, and these two are edited or modified plant by the genome editing technology, which deleted 620 nucleotide. And you can see almost 40 centimeter height has been reduced in the resultant variety. And this will be semi-dwarf version, has the ability to withstand in erratic climate, but with intact grain quality. This is how it could be precisely used to reduce height. Now I give you now taking you to a different technology, which is next generation genome editing tool. Because this CRISPR-Cas9 technology, this intellectual property is very, very complex because it is owned by mostly University of California, University of Vienna, Emmanuel Charpentier, Jennifer Doudna, Feng Zhang from MIT. So those people actually control the Cas9 IP, and you cannot apply them for commercial application. So you do not have freedom to operate. Yes, you can apply for your R&D in your laboratory part. You can publish paper, no issue. But you cannot commercially release a variety by using this technology until and unless you buy this technology license from them. That's very difficult. Even Indian government is suffering from that. Despite producing two ready-to-release variety using CRISPR-Cas9, now Indian government is negotiating with the license holder how to release it to the commercial market. Now, for that, what our lab was focused on to develop some alternative, and we have developed a very small version of genome editing tool, which is an alternative to Cas9 and Cas12a, which is the most popular genome editing tool that I just described to you, CRISPR-Cas9. So this was isolated. This protein was isolated from Deinococcus radiodurans, and you can see the size is 1/3. So So if this Cas9 looks like this kind of bulky in nature, and this is TNPB, very slim, fit. Okay, and this is an analogous one. Cas9 is 1400 amino acid like football, and this is baseball, which is only 400 amino acid. It means one third. This is TNPB. This one third of the size, so it makes it very easy to transfer into the cell. So delivery of the reagent become easy because of the smaller size. Now, let me give you some demonstration of what we have done in rice plant. So we have targeted a chlorophyll biosynthetic gene. Here you can see. And this gene is named HMBPP, and we have deleted 23 base pair in that gene. And as a result, the gene became functional. So the plant would not be able to produce chlorophyll any longer. And as a result, it produces albino plant. These whites are albino. Okay, they are no longer green. So it means this tool is very efficient in disrupting plants. This is how we have demonstrated there is a visible demonstration of this tool for genome editing. Now, this tool, as described by my introducer, this tool have been submitted to AdGene, which is a NGO that supply research material, especially plasmid vector, to different parts of the world, and this has been used by different part of the world, different laboratories. And this has got also several international attention by Europe, US, a lot of news portal. They have covered this development. And recently, I do not know if you have read in Indian Express in November 20, they also covered after we got the patent for this technology. So now we have the IP and we can use this tool for the developing commercial crop varieties without buying license from them. So this is how it is impacting Indian agriculture. Now coming to the second part, this is the first part that I talked about. It's to disrupt the gene function. Obviously, CRISPR-Cas9 I told you, and then next generation genome editing tool TNPB I told you. Transposon associated B protein, that's called TNPB. Now, this part would give you an idea how much it could do, different kind of technology derived from this original technology. Now, I already told you that how this double strand break happens, okay? But this postman can be made a little less functional, and one Caesar is absent now by making a mutant of this version, and this is called Nikkei's Cas9. Nikkei's Cas9 can do a single strand break instead of double strand break. So it does not have two Caesar, so it will only make a single nick in one of the strands. It's called Nikkei's Cas9. Another, it could be also made dead Cas9. Dead means it's not really dead, it's catalytically dead. What does it mean? It means if you give an address to the dead Cas9, it will go to the target sequence. It will bind there, but it will not make any double strand break because it does not have any Caesar in its pocket. Both the Caesar are dead, but it has the ability to bind, but not any ability to cleave the TM. So this is N-Cas9 and then D-Cas9 called dead Cas9. Okay, so why I'm saying this? It has reason. Because you can see here, with this kind of D-Cas9 or dead Cas9, you can fuse a protein that actually are known to repress transcription. So if you fuse D-Cas9 with this protein, which is a transcription repression, and if you give a genetic address, then this, suppose genetic address to the promoter of the gene, then this D-Cas9 craft fusion would go there to enhancer or promoter, it will bind there. And because of the availability of this repressor or inhibitor protein here in the promoter, the gene expression would be inhibited. And this is called CRISPR interference, which is different from RNA interference. This is called CRISPR-I. Now, another is called, similarly, you could use it to activate transcription of a gene. So what you need to do, you need to fuse a D-Cas9 with a VP64, which is an activator protein. Now, D-Cas9 bind with VP64. You give the genetic address to the promoter. Now, D-Cas9 will take the VP64 to the promoter. And as a result of lot of availability of activator protein here in the promoter, the expression of the gene would be increased in high amount. And this is called transcription activation. And this technology is called CRISPR-A or CRISPR activator. Now, similarly, what we have done, we made the TNPB in a dead version. And this dead version of TNPB was fused with an activator protein called VB128 and 6-XTAL. And then this is called TNP based activator. And when we have used it, you can see here, we targeted three promoter of the three genes in rice. And you can see this green color is the one that was treated with TNPB activator. And their expression is highly, almost tenfold higher than the basal expression of the gene, which is not treated. So this is what we have made, a miniature gene activator by using the TNPB tool that was discovered. Now, another technology is epigenome editing. Now, what is epigenome? Epigenome is, for example, the same gene is present, but in one genotype, it is highly functional gene, but in another genotype or another variety or another same species but different genotype, this gene is not functional, not active. But when suppose you do a molecular analysis and you see that this is because the promoter is methylated, it means this is an epigenomic silencer. So because our promoter is methylated, this structure is methyl group. The promoter is methylated, the gene is not functional or not getting expressed. Now what you can do, you fuse a D-Cas9 with a demethylase enzyme and give an address to the promoter. Okay, and then this is called epigenome editor. This epigenome editor will go to the promoter and it will remove the methyl group because of this demethylase enzyme, and the gene promoter would be demethylated, and as a result the gene would be highly expressed because of the demethylation. Now, without changing any letter in DNA, you are changing the chemical modification of that letter to increase the expression, and this is called epigenome editing. Now, this is the last part, not last, before the application, this is the most exciting part that I will tell you. And this was like, like science fiction just few years ago, like in 2016. Yes, 2016, first this technology came. So a few years ago, it was science fiction. Now it became real that you could cure genetic disease in in animals by using this precise editing technology. For example, let me give an example. So you all are Indian audience. So Indian audience, you should probably, most of you know this. This is a movie from Amitabh Bachchan. It's called Power movie. And can you name who is the character? What is the name of this character? Anyone? Anybody?
Odo is the name.
Yes, Oro. Thank you very much for your response. So Oro was suffering from a rare genetic disease called progeria. And now Oro's friend, here you can see, looks same and normal. They look so different. Oro is like 80 years old. And why? Because Oro is suffering from progeria disease, and that causes rapid childhood aging in children, and they normally die at the age of 14 or 15. And if you know the reason why Oro was suffering from this rare genetic disease, you would be amazed to know that this is because a single wrong DNA letter. Here you can see the healthy friend has a G letter, whereas Oro has a letter, which is a wrong letter. Now, because of the single wrong DNA letter, Oro was suffering from this disease and died at the age of 13. Similarly, if I want to give you a plant example, this plant is susceptible to a fungal disease. You can see all are dying because of the presence of a C nucleotide. And here it has T, and as a result, it became tolerant to the disease. So a single letter makes a disease-tolerant plant. Single letter change makes a suffering patient from a healthy patient. Now the question is there a way to rewrite genetic letters? Can we rewrite a genetic letters? Until this point, I have told you the technology to disrupt a gene function. A gene is functional, changing this gene to be non-functional, that is knockout. But is there a way to rewrite genetic letters? In order to cure RO, you need to rewrite A to G. In order to make disease resistance in plant, you need to write C to T. This is how you can do. But is there a way? Yes, there is a way. It's a technological base editors where you can rewrite a single DNA letter with another DNA letter. And this David Liu from MIT has actually invented this technology. Um, and he has won also breakthrough prize this year. So you can see with this technology, what you can do a lot of changes in the genome, very precisely A to G, T to C, C to G, and vice versa in the genome, and how it works in the genome. If I want to tell you, so what is the problem here? There is a problem in this genetic code is a wrong letter, so that's why in your sky, blue sky, there is no star. Now you have this correcting tool called base editing, and now your genome sequence is corrected, the gene is corrected, and now you get your phenotype back. This is how it works in the genome. Base editing, exactly. Okay. Now, if you have any difficulty, for example, difficulty in listening to my voice, just you can let me know, please, not me. Huh? So do not suffer from broken voice. Okay, so I think you got how base editing actually works in the genome. Now let me show you this video. So this orange color is a deaminase enzyme, and this is a Cas9. The single base pair is getting changed. So this is a single base can be changed. So let me give you an example from our laboratory. What do we have done? Here you can see here is N-Cas9. Now I have shown you a lot of example with D-Cas9, like date Cas9. Here is an example with Nikkase Cas9, which have a single nucleotide, a single strand break ability. This Nikkase Cas9 was fused with an deaminase called TAD-A, which is an adenosine deaminase. So it can deaminate adenine to inosine. And once you fuse with N-Cas9 this TAD-A, it makes adenine base editor, which has the ability to transform A into a G nucleotide in one of the strand, in the opposite strand T to C. But it has one limitation. Limitation is that your target A should be present between 4 to 8 nucleotide of the genetic address. Suppose you design a genetic address of 20 nucleotide. Out of this 20 nucleotide, your target A should be located from 4 to 8 nucleotide. So it means if it is outside of this window highlighted here, it would not be optimally edited. If it is only located here, it would be edited from A to G nucleotide. Let me give you an example here. You can see this was our target. This A is, what is the position of this target in the 20 nucleotide? 1, 2, 3, 4, 5, fifth nucleotide. It means it is within the activity window. And once this A was targeted with the base editor, and we targeted that, and you can see this A has been modified to a G nucleotide. This green color peak means it's an A nucleotide, and this black means it's a G nucleotide. And you can match all the others. You can match all the others changes, all the other peaks. Those are all similar. Only this A has been modified to a G nucleotide. Clear? Now, for another gene also we targeted, and you can see what is the position 1, 2, 3, 4, 5, 6, 7. Seventh A was targeted, and was also modified to a G nucleotide by the application of base editor. And we got, for example, in this case, B phenotype was the phenotype that we achieved because of this single nucleotide change in this gene called WSL5. Now, I will give you a very interesting example. So, in some cases, natural mutation is beneficial for crop improvement, but some cases, natural mutation could be also deleterious. For our purpose. For example, one natural mutation, what was natural mutation? 383 base pair deletion. That was the case for what? Green revolution, that we incorporated and made our plant shorter. Okay. That was the natural mutation. But in many cases, one single base pair change could make an appearance of early stop codon in the initial portion of the gene. For example, any stop codon or stop signal should be present at the end of a gene. But here, for one change, one letter, wrong letter, this TAG is a stop codon which is like a stop signal to the protein synthesizing machinery that appear at the beginning of the gene sequence. And as a result, this is called premature stop codon. And as a result, the gene cannot be produced enough. The protein cannot be produced in full length. That would give rise to a truncated version of the protein, and the plant would become susceptible to a disease because this protein is responsible for disease resistance. Now we tried to correct this stop codon with base editor. So we applied base editor, and you can see this T has been modified to a C nucleotide, the red modified to a blue, which is T modified to a C. And as a result, the stop signal now changed to a green signal for the protein synthesizing machinery because CAG is not a stop codon, and it will give rise to the protein synthesizing machinery to allow the full length synthesis of the protein. And as a result, the plant becomes disease resistant. So what does it mean? So we corrected the wrong letter with the right letter by the application of base editor, and we developed a blast disease resistant plant. This is how it could be applied for uh, disease resistance and other application. Now to introduce you to another advanced technological prime editing quickly. So now let me give you this another problem to you. What is this problem? This is called Tay-Sachs disease, and this Tay-Sachs disease, all the affected child dies at the age by 5. And you know why this Tay-Sachs disease happens? Because 4 extra DNA later actually inserted into the hexagene of this kind of child. Now, because now if I ask you how to cure this kind of child, now you would simply tell me, because all of you are very intelligent, very good teacher. So you tell me, okay, simply to correct this gene, we need to delete precisely this 4 DNA later. But if you delete 5 DNA later, the gene is again disrupted. You have to exactly delete 4 DNA later so that the gene become normal. Now, is there a tool you cannot do with base editing? You cannot do with normal knockout tool like CRISPR-Cas9 and other things. But now you have the tool called prime editing that I am going to introduce you, which allow you to do all this kind of modification. All 12 types of base substitution, small insertion, deletion, and combination, all these kind of things you can do. And what is it? It's a fusion of N-Cas9 here. You see the example, base editing uses N-Cas9. Prime editing uses N-Cas9. N-Cas9 fused with reverse transcriptase. And when N-Cas9 fused with reverse transcriptase, then it will call as prime editor, and that can do all this kind of things. Now, the genetic address is a little bit different that you have to add. This is a normal genetic address, normal guide RNA, how it looks like. But here is prime editing guide RNA or prime editing genetic address. It has 2 components in this. One is a search component. It means where to edit, in which portion of the genome, which chromosome, which gene, and at what position. This region of the address would determine search. And this region would be determined what to edit, what to incorporate, what to change, what to modify. That is coded by this region. So this is why it is called search and replace a genome editing tool called prime editing. Now let me give you an example. Here this wrong letter is 3 wrong letter inserted. So you have to delete this 3 and insert 1 right letter. You have prime editing tool and you can do this editing and you will get your phenotype back. This is how prime editing can be used for combination of edits like deletion and special insertion. Okay, now how it works. If you see in the genome, this is the search component where it will bind to the genome to fix the position where to edit, and single strand break will be made by N-Cas9, and then this replace component has 2 things. One is primer binding site, which have exact complementarity with the next strand, and it will bind with the next strand. And then this region is called template. Template means this is the region where you would incorporate what kind of edit you need. That's called template. And now this reverse transcriptase enzyme will reverse transcribe to read the template and incorporate directly into the genome by incorporating what is written in the template. It will copy like that, okay? Complementary base. So this is how it incorporates changes in the genome. Now, when we tried to change one of the target or nucleotide we wanted to change in plant, And we have tried with prime editing, but the first generation prime editor was very, very low efficiency, and as a result, we failed in achieving desired editing in 300 plants we have generated. But in that time we have done this reviewing of what others are doing in other part of the globe, and we have seen that a lot of groups from China, USA, they also showed that this technology has really low efficiency in plant, especially is very, very low efficiency. So what we have done, we have modified the editor by protein engineering, and we have developed improved prime editor version, and as a result we have now achieved to change one amino acid to another amino acid. So our target was arginine to glycine in one of the gene called phosphoenol pyruvate carboxylase, which is the gene which is very, very crucial for C4 photosynthetic pathway. Okay, so rice is a C3 photosynthetic pathway gene, but we wanted to see if we can modify this arginine to glycine so that we make rice Pepsi like a C4 version of Pepsi. So we were successful in modifying the C to G nucleotide, and as a result, the codon CGC changed to a GGC. Arginine modified to a glycine amino acid. And you would see the difference for the single changes in the genome. And in these cases, we got 7 to 1% editing efficiency, is a very, very high efficiency. And if I want to show you here, you can see this is wild type, which is not edited, and this is the edited one. What difference you can see? If you simply compare this plant and this plant has already flowered. This is not flowered. It means this is early flowered. So it is almost 15 days early flowered, the edited plants. It means you can save water, nutrition, and other resources, and even effort in taking care plants for 15 days if you can harvest your plant 15 days earlier. Okay. Now here you can see this is an extraordinary symptom that we have seen, that wild type has light green leaf and the edited plant has dark green leaf. It means it is an indication that chlorophyll is higher. So you can see we have measured the chlorophyll content of this control and edited plant, and you can see highly significant difference in chlorophyll content, and as a result of this high chlorophyll, it has also significantly higher photosynthesis. Now we could increase the photosynthetic efficiency of the C3 rice by changing that single nucleotide. Now these are other impact that we have not expected. You can see here the 10, in all cases, 10 seeds are arranged, and you can see wild type seeds length and breadth both are increased. So this is the 10 seed, and you can see 10 seed crosses this line in case of edited. 10 seed crosses this red line in case of edited. It means both length and breadth have been increased for this single nucleotide chain. And total 1,000 seeds weight also increased significantly because of this larger enhancement. Now, when this clue we got that is yes wild type and here is edited one. So it's a chlorophyll more accumulation that's already proved by our analysis. Now, when chlorophyll biosynthesis is lot dependent on iron availability and iron transport into the plant cell, so then we thought that probably there is some iron changes in the in the edited plant. And what we have seen strikingly, that edited line here is a staining. Dark blue means more iron. You can see edited seeds has more iron in it, and wild type has less iron. So it means we can now biofortify. So this iron would more iron available in seed, and that means it could help us to alleviate the problem of iron deficiency in Indian women. So that's a great objective in crop biofortification that could be fulfilled with this plant. Now, similarly, as we know that iron and zinc are co-transported in plant, then we have also done zinc analysis, and you can see edited plants. This 4 are from edited, this 5 are from edited, and this one wild type. And all 5 plants showed higher zinc accumulation. They also showed higher protein accumulation. So it's a high zinc, iron, and protein accumulation. They are nutritionally superior. So we have never thought that this would happen. So this is an accidental discovery that we came up. Now, I would soon publish this article, and then you can read more about this. I'm not going into the details. But this question to you, why the whole world are interested on plant genome editing? Why not plant genetic engineering, transgenic technology that came earlier? It is for this reason. You can develop a beneficial trait without permanently incorporating foreign DNA. So no foreign DNA need to be present in, but you can develop a trait. How it is possible? Let us give you an example. For example, you are applying base editor. So here is the DNA. Base editor plasmid is incorporated in chromosome 1, for example. For example, now this base editor plasmid will produce a protein, base editor protein here. This base editor protein will go to the target. The target is in chromosome 4, for example, because the address you have given for chromosome 4. Now this target is now modified. The DNA is modified by shown here with asterisks. Now, if I ask you, once the DNA is modified, do you think this base editor yellow color protein, DNA is any longer needed in the cell? Anybody, yes or no? Once the DNA target DNA is modified, do you think this base editor yellow color is needed any longer to be present in your cell? Yes or no?
No.
No, yes, because this is already done. The target modification is done. Perfect, thank you. Now, what you can do through simple Mendelian segregation, 3:1 segregation would happen. If you grow to the next generation, all the sexually propagating plants. Now, you could generate only target modified, but without any base editor integrated. And that is why you can generate your mutant without any foreign DNA. Here, what foreign DNA you have used? You have used base editor foreign DNA. You can just segregate out in the next generation, and you can obtain a mutant without any foreign DNA integrated by simple sexual segregation. So that it's not any foreign DNA is not present, but you get your trait developed already by the application of the base editor in the first generation. So Mendelian segregation causes this. Okay, clear? Now this is the last part. Quickly, I'd finish in 5 minutes probably. Gene editing applications, and that will probably give you some goosebumps. Those who are not exposed to, but those are already exposed, they already know this. Okay, so what is this? Let me give you an example. Simple. These are the examples. Field that has already been demonstrated in agriculture. It's not a potential example. This has already been developed. Yield, disease resistance, climate adaptation, taste enhancement, nutritional quality, storage, all have been done in agricultural development by applying these technologies. Now, this technology, those who are important in disease resistance and plant defense, you can see these are the plants and these are the pathogens. There are a lot of plants, diverse group of pathogens. Those plants have been modified with this technology to develop resistance against those diverse group of pathogens, be it bacteria, viral or fungal pathogens, even parasitic plant. So you could go through this review article to get a glimpse of how this is actually, revolutionizing this field also. Now, crop domestication, I have started my lecture with crop domestication in the wild crop to cultivated crop, and that took a lot of time. Like, for example, thousands of years of selection, natural mutation, selection, natural mutation, selection. But nowadays with genome editing, you could do a cultivated crop, a wild crop modified to a cultivated crop very quickly within 2 to 3 years, even 4 years, within 4 years generation. That's called accelerated crop domestication. Quickly crop domestication you could do by targeting a specific gene that are responsible for wild character. So you can disrupt those genes, and as a result, wild character would be gone, and they would look like cultivated crop. That's called crop domestication. And this is, you should remember that these are the crop that have already been in the market all over the world. For example, soybean edited in the US, which has an oleic acid 80%, which is like olive oil composition. Soybean oil, but olive oil composition has been made possible with genome editing. That's available in the US in 2019. Japan 2021, this is a high GABA tomato that help to low blood pressure. GABA is a, non-amino acid protein, non-protein amino acid that reduces hypertension. That tomato is available in Japan. If you go to Japan, you can have it. This mustard green means mustard leaf with low pungency has also been developed to be used in salad in the US. That is also available in several markets like Walmart, Ways, even if you go to California. And these two fishes are also available in the Japan, which have been edited and commercially available. Now, this is a field I have taken. This has given by Dan Waters, who has developed this in the US. So Dan Waters' photo I have shown you. This is Dan Waters has developed this soybean. This is the genome edited soybean, which the composition of the oil is like olive oil. This is a field from Minnesota. Uh, this is a huge. This is from 2019. Now, this is Victoria Gray, the first patient who has been treated very successfully for sickle cell disease. So, sickle cell anemia and beta thalassemia are the two very well-known genetic diseases. We read from our biology days, from 8th standard, or maybe now it is 5 or 6 standard. So this therapy is called Casgevy, which is a CRISPR-derived therapy. These are already available, first commercialized in UK, and then it came to US and other countries. Now these are the countries where this therapy is available in clinics. So if you know somebody is suffering from sickle cell disease or beta thalassemia, you could tell them that to go there and get it treated. Okay, and also for your good example, from this month onward, also India is also starting clinical trials by our own technology developed from CSIR IGIB in New Delhi, Devajyoti Chakraborty, for sickle cell disease. But this is already available. You can get it treated from those countries. But okay, so as I showed you the example of progeria, you can see this is a mice affected with progeria disease. This is a progeria mice model. Now this is treated with base editor. So you can see this base editor progeria affected mice has been already treated successfully in a model, and now it would come into clinical after clinical trials. So this is how it is really modifying. Now over 300 clinical trials are undergoing all over the world for different HIV, different type of cancer. Now let me give you an example of the treated patient called Alisa. Alisa was suffering from leukemia. And Alisa was 13 years old, and Alisa has been treated with base editing therapy. And this year in 2025, now Alisa looks like this. And she was invited to, during the award ceremony of Breakthrough Prize. So Breakthrough Prize like an Oscar of science. So this has given to David Liu, as I showed you the photos earlier, the inventor of this technology. And now Alisa said in 2025 that I have now a future, which I have never thought that I would have a future. Now she is already cured with this base editing technology, leukemia, blood cancer. Now this is some crazy example or crazy application of this technology. You can see bringing back of woolly mammoth. So they have edited 5 genes in a mice model, and you can see their fur looks like an woolly mammoth fur, like lot of resemblance. This is obviously first step, but they're trying to do, and that company is called Colossal Bioscience. If you want to read more, you can go. So what I showed you, a tremendous potential of this technology. Now, tremendous potential of this technology. Now, when there is tremendous potential, there is tremendous risk also involved. When this first recombinant DNA technology came into force, when recombinant DNA technology came into force, that time people are very, very, skeptic what would happen with this technology. It could be used for many bad purposes. And that time in 1975, first Acylomer conference was convened in California, in the Acylomer group. And that was the beginning of regulating recombinant DNA technology. Whatever the regulation that you see for recombinant DNA technology, genetic modifications, that came out of this conference in 1975, organized by Paul Berg, Stanley Cohen, Boyer, those are the, and Watson, those are the organizer of the conference. And that output of that conference, 3 days conference, actually adopted by all over the world as a guiding principle to regulate this cloning technology. But after 50th anniversary, in the same venue, same date, this year, 2025 February, because of the emergence of artificial intelligence, gene editing, synthetic biology, mirror biology, Mirror biology, if you know, and chemistry people should know, mirror biology is that all the biological amino acid and sugar could also be made in a mirror form, like T form and L form, opposite form. And if an organism and bacteria developed with those kind of mirror isomers, then those bacteria could not be detected by our immune system. So that could be a huge problem for a human being. So that's why, because of the emergence of these highly powerful technologies, so organizers have thought that we need another asylum at this time because of this technology. And there was a Spirit of Asylum organized this year in February. And I was very lucky to be invited as one of the next generation leaders. And I was only invited from India. And this is, Craig Venter, and there are a lot of people like Cohen, Boyer, and other Nobel laureates also attended. But I just shared one photograph. This is Craig Venter, who has actually done the genome sequence of human genome sequence. He is the pioneer, Craig Venter. Okay, so what I'm saying, I would recommend you to go through this article we have recently interviewed in published in Trends in Biotech. So how this next era of biotechnology should be governed because of this availability of these technologies. So it's not me, although I was the lead contributor, but there are other people from different part of the world. They have also, they are expert in different fields. They have given their ideas how to govern next era of biotechnology. So to make it easy for you, this is a protocol book that we have edited, as mentioned also during introduction. So you can go through these books to use in your research if you want. These are protocol books, not a simple book chapter kind of things. This is stepwise protocol material methods are given to adopt this technology. Now, to apply this technology for your experimentation, there are a lot of computational tools. And there are a huge number of computational tools, but for which purpose, which computational tool to be used, our people are very confused. So that's why we made this repository and recently published in this channel. It's called CRISPR-Gate, and CRISPR-Gate is a repository where you can go and select. You need to design a guide RNA for base editing. So what are the tools to be used? And there is ranking of the tool, web tool that you can. This is a great resources for the new researcher from the global south. Okay, so this is the last slide. So I must acknowledge that I have been trained in University of Calcutta by Professor Satandra and Dr. Parveen. We learned genome engineering, and I did my Fulbright postdoc 2 years at Penn State with Professor Inong Yang, and this is my current groups, although some of them left and new people have already joined. And these are the funding agencies that I use fund to run my lab. And thank you very much for listening to me, listening to you. And James Watson is no longer, because just a few months ago, I think 1 month ago, he has died. And this is James Watson. I was fortunate to meet him in Cold Spring Harbor Laboratory. Thank you very much, and over to you, myself. And I should stop sharing.
Not only enchanting your talk, not you just ended in perfect time. Your precision is accurate, just like CRISPR-Cas, and I must say it was really we were really awestricken. The way you have explained the new era of genomic editing. Now it's a time to invite questions from our listeners. Please ask questions, clear your doubts, so that ultimately we get more knowledge from this great person. Already, I think one question has been posted. It is from Dr. Just a minute. Dr. Prajit Nambiar. Excellent lecture. Mm, what, sir? It's a rice variety with GGC codon change. Even though the plant flowered early, it looked shorter than wild type. Does it have to do with anything with gene editing? Acceptance of GMO crops in society is still controversial. Are there any measures taken to generate awareness?
Okay, that's a great question, dear Dr. Rajit Nambiar. So, yes, you're right to observe this short height. So, yes, we have seen percentage of height reduction, edited plant. So, yes, edited plant was height reduction is one of the pleiotropic effect that we have seen. But in many cases, height reduction is beneficial, but we do not know whether this height is beneficial, because we have generated in T2 up to T3, we are growing them. Height is reduced, but all the yield parameters are intact. Even I show you the grain number, the grain size has increased. So yes, it has height reduction, but height reduction does not have any agronomic impact. Means in the yield, it does not have any impact. So we are mimicking this for other varieties, which is tall, and which is also farmer want to make them reduce. So we have to see whether in other variety these changes also reduces height, and if that height reduction also beneficial for farmers. So you're right. And then another question is GMO. So this genome edited crop are not treated as GMO all over the world. Mostly, including this, just past month also European Parliament also have decided to deregulate this GMO, not the gene edited crop, not as GMO. So for example, if I want to give you an example from India. So India in 2022 May, India has taken a decision that gene edited crop that does not have any foreign gene integrated in it should be treated like a normal crossbreeding cultivar, not as GMO. So it should not have, it would not have any necessary to go through stringent regulation or taking permission from the government if it does not have DNA in it. So that's why it is already deregulated and not treated as GMO. That's a good thing. And obviously there are a lot of this lecture also kind of honest. Obviously all of you are teachers, and my voice is amplified by 40 or 50 45 other your voice. So you go to your class and you tell this amazing technology, and it's kind of mimicking natural variation, and that's the voice amplification, that's the awareness actually I'm also doing. So yeah, thank you for pointing out this.
There is another question from Dr. Namrata Ray. Her question is there any way to eliminate NHEG, non-homologous recombination with HDR? Homologous way of recombination can take place during DNA repair to eliminate unwanted errors during editing.
Okay, that's a great question. So, yes, in HEJ, in higher eukaryotes, if you see like animal, plant, those are all higher eukaryotes other than bacteria and fungus. So those higher eukaryotes has a very high efficiency of NHEJ, and HDR is very less efficiency. It's a meager efficiency. Okay, Dr. Namrata. So that's why performing HDR in higher eukaryotes is really difficult. Although in animal it's little easier than plant, but it's still very, very less efficient, like within 5% efficiency. But if you do an NHEG editing, it's like more than 80% efficiency. So that's why HDR is commonly not recommended. And now, because this base editing and prime editing came, now HDR is highly replaced, because HDR allow you, because HDR has the highest possible potential. HDR can be used to do any kind of editing, because you have to supply a donor template, and that donor template should harbor those editing that you want. So actually, whatever combination you want, you can incorporate. But the problem is the efficiency. And that's why now this base editing, prime editing allow you to do some of those combination of editing, precise editing. And now people are very, very interested for base and prime editing, but not HDR. But many people also in certain cell type, HDR also shows good efficiency. In that cases, people take risk of doing HDR.
Okay. So thank you, sir. I just wanted to point out, congratulate you for the TNPB enzyme, which allows us to do genetic editing without any foreign license. That's a great deal. And sir, also congratulations that you have met all the flag bearers who are working in this field. You have met Watson, who discovered the double helical structure. You have met Doduna, you have met David Liu, all of who are doing, the cream research in this area. So great work, sir. Please continue to do so and inspire us. Thank you.
Thank you very much for inspiring with your words, Dr.
There is another question from Dr. Shomabu Chatterjee. Sir, is there any epigenetic influence work on gene editing or in its recombinants?
Epigenetic influence work on genetic. Okay, so probably if I understood clearly, Dr. Chatterjee, so your question is if we do gene editing, is there any other epigenetic changes also happens? Is that your question? And if this is your question, for example, so there is no because
Dr.
query
Chatterjee,
is
you can directly
whether,
directly have a talk with sir.
yes, yeah,
whether
yeah,
there is
please.
any epigenetic influence work on the edited genome or the recombinants, or you are
Oh yes, yes.
suppose you are designing for certain outcome of phenotypes, whether any epigenetic other influences actually cover up or mask that, or any kind of challenges is observed.
Yeah, yeah, you're right.
That
You're
is,
right. You're right. Yeah, I got your question. Thank you. Yes, so there are some studies that have studied that yes, this is our edited plant and this is control plant now, and these two plant edited and control are compared for their epigenetic status, and it's already published. There are several papers, and that shows that there is no significant difference between an edited versus a control. So that's why it does not have any impact. This is one aspect of your question. I hope I have answered your question. Okay, so next question, I think Dr. Rakhi
Raki
Chakraborty.
Chakraborty, yeah.
Yes.
Does gene editing edited rice plants show reduced heavy metal uptake and accumulation?
Okay, so reduced heavy metal uptake and accumulation. I think your question is specific to the work that we have done with that codon change in this Pepsi gene. So we have not yet analyzed heavy metal because it's a new work and we recently filed a patent and we're going to publish soon it is. But we are checking for, like, for example, aluminium and cadmium also. uh, and we have to see. I do not have a black and white answer whether it has means increased or reduced accumulation. I do not have an answer.
We can
Yes, sir.
invite more
Thank you.
questions. We have already got 2 to 3 minutes extra, so we can invite another 5 minutes we have in hand. So
Yeah.
we can
So,
ask the
hello,
question.
Dr. Kutub. Thanks for the exciting lecture. Yeah, I have one question regarding your TNPB. So, is it equally effective in the dicot system also?
Oh yes, I have. Yeah, if you read our paper, we have also demonstrated in Arabidopsis, and yeah, we just demonstrated that it works in Arabidopsis. And there are several later work published by other groups after our publication. They have shown in tobacco and independently in Arabidopsis also, so it works in dicot very well.
Okay. And regarding the editing efficiency, how much it is comparable with normal Cas9?
So you see editing
SPCas9?
efficiency, yeah, side by side you cannot compare because the targeting region of TNPB and Cas9 are not equal because Cas9 needs a PAM sequence which is NGG and TNPB needs a TAM sequence which is TTGAT. So it's a little different region, but if you compare like same exon or same gene,
Yeah.
then it is comparable. In some cases, TNPB showed better efficiency. In some cases, Cas9 showed better efficiency. So it depends on the target to target. So in one cases, we got 69% efficiency, so very high efficiency. In another cases, we get like 12% efficiency. So it depends.
And what about the off-target effects?
Oh, it has less off target because the PAM is stringent because TTGAT is a bigger PAM, TTGAT, than the smaller PAM NGG. So that's why it is less off target prone than the Cas9.
Okay, okay. Thank you so much.
Thank you. Yeah, I know you, Dr. Ram.
If is there any other questions, so we can directly ask sir, or our participants can just type in their questions. Uh, since a student of plant biology, may I ask you a question?
Yeah, sure, sir. It would be pleasure.
It's my pleasure. Sir, is conventional breeding technique, is it taking a backseat because so much of advancement in the gene editing technologies? Nowadays, we used to read our classical books that it takes 5-7 years to rather prepare a proper clone line for the classical breeding programs, and the environment also plays a greater role in the making the architecture of a genome. So somehow, if the gene editing is done, it's giving first generation or second generation, it's giving the best results. But what after 5 or 6 years down the line, in the field, when it's coming to the commercial production level,
Yeah.
is there any chance of getting the gene altered or the base altered because of the natural influences? Can that be minimized?
Yeah, it's a very good question. So yes, environment has a great role in shaping our plant architecture, even the genome architecture, as I also mentioned. So normally when you are creating a mutation, that is normally whatever application we are doing now, normally mimicking a natural variation means we know that this genetic variation actually helps the plant to resist a disease or to reduce the height or yield better, and we know from nature already, and we are mimicking that to our local culture. Okay, so it means those natural variation already existing in some genotype in some part of the world already for long time, and there is no elimination of that genetic variation. Now it is being created very specifically with genome editing, so it is less likely that that genetic variation would go away. Now, for example, if I for debating, if you just tell me that, okay, what if after 5 years I see that out of 100 plants you get like if it is a fixed one out of 100 plants, no plant should have should give away that genetic variation. But suppose, for example, you are saying that, okay, there are 5 plants that does not have that genetic variation that you created with genome editing, but yes, there are 95 plants. So that 5 plant would not be selected by farmers. They will be discarded, and the seed from that plant would not be used. Only the plant that are giving the trait, the seed would be collected from these plants. So it would be automatically selected by the users, the genetic variation and the resultant phenotypes. So that's why it would be stable in nature. There is no theoretical remote possibility of going out that genetic variation also.
Thanks a lot, sir, for clearing my doubt. So we are just at right time.
Yeah.
So uh, before I wrap up the whole session, let me give my heartiest congratulations to Dr. Kormolla for his groundbreaking research in the field of genome editing. What we actually teach our classes, he has demonstrated through his visuals lively in front of us. Thank you very much, sir. Your caliber, your benevolent work, and side by side, your intelligentsia would definitely enrich Indian agricultural research scenario and definitely revamp our concepts in coming future. So before I conclude, on behalf of the organizing committee, again, I would like to convey my best regards and best wishes to you. And definitely in near future, we would like to invite you again for your groundbreaking research, making our vistas open for newer concepts. Thank you all, sir. Thank you, participants, for your patient hearing and question answer sessions. Uh, Now I'm concluding the session for this time. The next session will start at 2 o'clock. Thanks a lot.
Okay, so quickly before you log out, so I would share with you some resources to share with the participant, and so that they would be benefited with that. So, and you can circulate to them. Okay, thank you very much for your time, and I
Thanks a
highly
lot, sir.
appreciate it.
Thanks a lot. Thanks
Thank
a
you
lot.
so much.
Now it's lunch session.